Morning, everyone. In the interest of time, I'm going to just start the session with some information. So please remember that the Wednesday is the sort of exciting day, that there's lots of moving parts. Okay, so there's a parallel session, there's a poster session, there's a game drive, and we're eating somewhere else tonight. So just to take you through that, we'll be having the parallel sessions. This session will be recorded and it will also be uploaded afterwards. I can't have two live at the same time, but this one will also be uploaded for people because those of us that have FOMO, like myself, and that want to be in both sessions can then do the other session afterwards. So we've only had a parallel session, I think, once before, and that was way in the Star Village at the so this time it works much easier because it's right next to each other, and we are actually going to allow people, if they want to move between the two sessions, to do that. So if you're going to be one of those floaters, please sit in the back and please just respectfully, leave and come back in. That also means that we will have to be quite strict on the time, which I'm thinking is probably not a good idea to have me as the chairperson. But anyway, we're gonna try our best to stick not just to the time allocated, but to the physical time, like 8:15, etc., to facilitate that. So we'll be having the sessions, and then this afternoon, straight after this, there's going to be the poster session, and the poster session is here in the foyer, and there will be wine and there will be eats. Okay, so entrees, I suppose. So please stay for that. We'll finish probably around 4 o'clock here, and then the poster session will be in here for 2 hours, and then at 6 o'clock. The game drive vehicles will leave from here, and we will be going on a 1-hour night drive from here. And then those vehicles will take you directly to the golf club, where we will be having dinner. And then from there, you will be coming back here again on the game drive vehicles. And then those of you that stay outside in other accommodation with friends or in the research camp, you will just have to show this tag to get out of the camp to your accommodation. Okay, so essentially just don't lose the pack. Okay, everything will be happening here, and then you will be going away from here, and you will be taken care of, fed twice, and then come back here, and then you can go to your own accommodation. Okay, so in the interest of time, let's kick off with the session. So the session has got a lot of interesting ecology, modeling, remote sensing, techniques, technology, data. So it's quite a varied session. I think it's going to tick a lot of boxes. So it's great to have you all here. I know it's a bit confusing with two screens, so just pick a screen, but pick both screens in between so that everyone gets a chance to see you. And we're going to kick off with Edward Mohoku on phenology.
Good morning, everyone. I'm Edward, a PhD student at the University of Bayreuth, and I'm glad to give you a talk on changes in phenology. There's an overwhelming amount of evidence which suggests that climate change is impacting the functioning of terrestrial ecosystems, which is evident in shifts in species distribution and changes in phenology. Vegetation phenology plays an important role in regulating water, energy, and carbon feedbacks in the biosphere. So scientists study changes in phenology to gain a deeper understanding in terms of how our environment is changing over time. Based on that, they then develop response measures like early warning system, proactive adaptation strategies, and mitigation and adaptation measures. One would ask, why Southern Africa? Well, there's a lot of literature which suggests that the region is one of the most vulnerable to climate change, and I think there's a lot of literature. Can I use that? There's a lot of evidence that suggests that it's one of the most vulnerable regions to climate change, and it's also going to be further impacted by future climate change. So we are trying to understand whether we are already experiencing such impacts. Well, we are terrified that whenever we study changes in phenology, we are actually reporting changes in land use. So we chose protected areas to have a reasonable level of understanding that the changes that we are detecting are actually related to the climate system rather than a farmer somewhere else increasing fertilizer on their field, which will eventually affect the signal that we are detecting. So in this study, we are addressing two questions. The first question is to understand the patterns of changes in phenology. The second question is to understand the nature and the magnitude. By magnitude, we are referring to how widespread are these changes are. Where are the changes most severe? Which are the changes, which are the areas that we may regard as phenological change hotspots? So to do that, we use EVI. I think most of you have probably heard of NDVI, which has been heavily used in remote sensing to study changes in leaf phenology. NDVI is a normalized difference vegetation index. It's essentially a ratio between the near infrared reflectance and the red reflectance. So it's essentially a proxy for the amount of light energy a leaf is absorbing. The more chlorophyll a leaf has, the more light it absorbs. But one of the problems with NDVI is that it tends to saturate at moderate level of vegetation cover, and also it's sensitive to atmospheric and soil-induced noise. But it has also been shown that EVI tend to produce a cleaner signal compared to the two. But basically, EVI also is meant to show vegetation reflectance. The study region has a pronounced level of climatic seasonality, which then tend to affect vegetation phenology. So this is an animation of 4 years showing time changes in seasonality in the region. I'm running it at a faster pace, but it's difficult to see whether the changes are ongoing, but they are ongoing. The next slide also shows the same animation, but just basically for 1 year, this time at a slower pace. This is just to show you that climate has divided this region into 2 seasons. One is the wet season, which usually begins in October up to April, characterized by high productivity, high growth rates, high reproduction. The other season is the dry season, which is characterized by dormancy where plants close their stomata to conserve water. We are adopting an approach by Bettenwerf and colleagues, and we begin with our approach by fitting a cubic spline to the data sets over a 20-year period per pixel. We are doing this per pixel. So from the spline, we are extract 21 ecological metrics that represents the life cycles of a plant from the start of the growing season to the end of a growing season. But we then have a problem. Our study area consists of ecosystems such as desert, savanna, forest. So this therefore means that the phenological signal differs in a desert compared to a savanna compared to a desert. So we risk diluting our signal of change that we are detecting if we do not separate this region into phenological group based on the similarity of the pixels. Pixels that belong to one another should be compared to one another, and pixels that don't belong to one another should be separated from one another. So to counter for that, we group our study areas into phenological group we call phenoms by applying an unsupervised clustering of the phenological data. Further on, we split the data set to enable change detection into 2 parts of the record. And then we assess change based on these phenoms by
normalizing such that the mean is 0 and the standard deviation is 1. This is to obtain a comparable measure of change from one place to another. Okay, this is just basically a repetition of the spline that we're fitting to the data, but I just wanted to show you a schematic diagram of what I'm talking about. On the y-axis is your EVI data set. I will interchangeably use it together with EVI intensity, but basically I'm just talking about the same thing. And on your x-axis is just your time, your phenological year. These are day of the year from 0 to 365. In red is the spline that we are fitting to the EVI point data that we've extracted per pixel. We are then extracting phenological metrics. Actually, there are 21 of them, but I'm just showing you a couple of them. So our metrics have 2 dimensions. One, EVI value, and another is a time value. Some metrics only have 1 dimension, either a time value or an EVI value. For instance, the metric peak is a 2-dimensional metric because from it you can extract an EVI value. You can also extract the timing or the day at which the peak occurred. The same applies for the start metrics. Also the same applies for the end. But metrics such as GSL, which is just the number of days between the start of the growing season to the end of the growing season, is just a time dimension because it's just a number of days. The metric like amplitude is just the difference between the trough and the peak, which is an EVI value. So some metrics have X values, some metrics have Y, and some have both. So by creating phenological groups, this enables us to obtain an average signal between the first period and an average phenological signal also in the second period. So this makes us to study the dynamics between the 2 periods. We are also be able to show, because we have 2-dimensional metrics and also 1-dimensional metrics, we are able to create graphs that can display both dimensions in space in terms of vectors. So for instance, if you assume the matrix from T0 to the diagonal T1, and unfortunately I don't have a pointer, but the diagonal metric at T1, it just shows that that metric has changed from T0 to T1 diagonally. So in that, it is a 2-dimensional metric because you are able to obtain a time dimension that has changed. So from T0 to T1, it suggests that the metric is occurring later, while from T0 in the y-axis, it suggests that the metric has increased. So it can also occur in different directions, whether down or up. But some metrics that are measured in time dimension, they can only change horizontally. So from T0 to T1, it means that metric has been delayed, while some metric with 1 dimension can only change vertically from T0 to T1. So, but I think it will be much clearer when we use real data to produce the plots. So this is our output from the cluster analysis. These are regions with similar phenological properties. It's not necessarily a vegetation map or a biome map. It's just, in principle, a map consisting of metrics with similar phenological properties. These metrics are timing metrics and also metrics in EVI metrics. So the next slide, we are just going to concentrate on one of the groups, which is group 7, just to further disentangle or dissect the changes in this group. So I think if we can recall, we saw an average phenological signal in red. This is basically showing the same thing. And also, we are also seeing the vectors of change metrics. In green are the start of the growing season metrics, while in red are the end of the growing season metrics. We can see that the start of the growing season metrics have occurred earlier, while the end of the growing season metrics in red have occurred later. This translates that the growing season in this metric, in this phenome has been extended, as can be seen by the blue matrix. We were also able to show total change in the phenological behavior in terms of their magnitude of change. This enables us to show potential areas where change hotspots exist And one could also infer or interpret it as such that this could be areas where rates of phenological change are either being exacerbated or rates of phenological change are being reduced. Because we are dealing with metrics with 2 dimensions, we are able to separate between the two to enable us to tell whether these changes that we are seeing, is it really being driven by EVR metrics or is it really being driven by timing metrics? But we can see that most of these changes, the EVR metrics are more dominant compared to the timing metrics. So just to sum it all up, we've shown that all our metrics are undergoing some form of ecological change. Changes in the growing season are playing a more dominant role compared to other metrics. And changes in the EVR metrics are more widespread. A possible explanation for this could be that there's recent literature that suggests that the region is getting warmer. Rainfall is becoming less seasonal, therefore translating into an extension of the growing season. But some drivers such as CO2 fertilization cannot also be ignored, which have been shown to increase water use efficiency in plants and thus extending the length of the growing season. Some drivers as well, such as fire and herbivory, should also be taken into account. So our main finding is such that these changes are likely to be driven by changes in the climate system. So we have shown that relevant ecological changes are ongoing, but there still need to be a synergy between ground-based studies and remote sensing-based studies so that ground-based studies can be used to validate these remote sensing-based studies to improve the accuracy in terms of our estimates. We have shown, this is just a change detection study, so we have shown where the changes are occurring, when the changes occurred, and how they've occurred, but we didn't say what caused these changes. So there needs to be an attribution of directly linking the drivers of these changes, which will then fall into an integrated monitoring system for the Southern African region, which at the moment doesn't exist. So with that in short, I would like to acknowledge the chair, staff members and the chair of plant ecology for their academic support and the DAD for the financial support. With that in short, I thank you.
Thanks, Edward, for a great talk. We've got 1 minute for one quick question. Corley.
Thanks, Edward. That was very interesting. And I was wondering if you separated the Mediterranean systems, you know, the ones you find in the Cape, from the more tropical ones, and whether there's a difference in the patterns. I think, from what I hear, they feel that the Mediterranean, the rainfall season has shifted, so not necessarily longer, but, you know, arriving later and staying later.
Yes. Basically, we didn't look at a Mediterranean is basically a biome, right? Like part of the fynbos biome. We are basically looking at phenological properties that are similar to one another. We are not looking at ecosystem level. We're just looking at the fact that the pixel properties from the phenological property of a pixel from this pixel to the other ones. We need to group them to one another before we assess changes into one another. By doing that, it avoids us from comparing mangoes to oranges, because the signal in the fynbos or the Mediterranean, which is occurring probably the wet season is in winter, compared to the wet season in the desert or the savanna, is totally different. So the signal will be different from the Mediterranean, and in the savanna will also be different. So we are not really looking at it at biome level or at a vegetation context level. We are just using purely reflectance patterns from the EVI signal.
Thanks, Edward. We're going to go on to the next presentation, which is Steve Higgins, and he'll continuing on the theme of time detecting change.
And so, if the slides will appear, I'm going to be talking about detecting and attributing change in savanna ecosystems. This is work that I've done together with Timo Conradi, who's back in Bayreuth, and Edward, who's just given a presentation. So when we look at a picture like this, we naturally ask ourselves, are savannas on climate system-driven trajectories of change? And you can look at these sort of anecdotal images and convince yourself of different things, or you can also try and analyze it slightly more systematically. And when you do a more systematic study, what you find is that there are some regions where we're seeing increases in tree cover, some regions where we're seeing decreases in tree cover. And so the picture is more nuanced, and some of the rates of increase and the rates of decrease are also quite variable in space. Here's another example of a similar study using a slightly different methodology, and we're again seeing some regions where we're seeing increases in tree cover, other regions where we're seeing decreases in tree cover. So when you, obviously as a savanna ecologist, you're interested in trying to understand what's going on, you can unpack your favorite list of environmental drivers for savanna ecosystems. So you might come up with a list like this. This is my list. You might have a different one: fire, photosynthetically active radiation, atmospheric CO2, air temperature, herbivory, soil temperature, moisture levels in the soil, and then, well, I guess here we'll probably diverge quite a bit, your favorite list of nutrients, nitrogen and phosphorus, for example. So you could say, well, are these factors and changes in these factors driving the changes in the ecosystem properties that we are seeing? And of course, we then realize that this attribution business is kind of difficult. One of the reasons is illustrated by this study is that the potential environmental drivers that are forcing these changes they co-vary with one another, so they're highly correlated with one another, which means that isolating which of these potential drivers is actually causing the change is difficult. So fortunately, here comes my physics envy. The climate scientists have kind of shown us how to do this. So, I mean, the simple narrative of this is they observed something was going on in the atmosphere. They noticed that the air temperature was increasing. They wanted to understand what is causing that. They built a process-based model capable of reproducing that pattern. And then they essentially do a sensitivity analysis where they say which one of the drivers in that model is responsible for producing this pattern, in this case the increased air temperature. And what they did was they removed the anthropogenic drivers from that model, so things like CO2 or greenhouse gas emissions and also land use change. And when they do that, you get the little green lines at the bottom, and you're no longer able to reproduce those increases in temperature. So this we can call the detection and attribution workflow, and we were kind of interested in seeing whether we could apply something like this to savanna ecosystems or to ecosystems in general. So the first step is detection. We could say this is what ecologists do all the time. We're pretty good at observing and quantifying changes in ecosystem dynamics. The second step, which I've called the calibration step, is essentially to use an all drivers included model to simulate the ecosystem dynamics, and this model needs to be capable of also reproducing the aspects of the ecosystem dynamic that you're actually interested in understanding. And then the third step is the attribution, where you essentially do a sensitivity analysis and you kind of declare attribution when removal of driver trends significantly diminishes the ability of the model to reproduce the ecosystem's dynamics. So what we do in this study was we're looking at NDVI. Edward told you that NDVI is not the greatest, but it's got the longest time series. So we looked at NDVI as an index of ecosystem behavior, and this is a time series from around 1982 to 2015. And when you look at this pattern, it's pretty difficult to see if there's anything going on in that time series. So what we tend to do is we decompose that time series into component parts. It's helpful as a first step to remove the seasonality in the time series, and then to then look at whether there's any long-term trend once you've taken the seasonality effects away. Once you've done that, you might be still left with some residual variance, which is essentially unexplained. So what one is doing is essentially an anomaly analysis. So here we've removed the seasonal trends, and then we're looking at the NDVI anomalies, the deviation from the long-term means. And so you can plot that, and then you can try and fit a trend line through that. In this case, there's many ways to skin a cat. There's many different kinds of trend lines that we could fit. And in this example, we've used the bent cable regression method. And this shows that in the initial part of the time series, we're seeing an increase in NDVI, and then in the second part of the time series, we're seeing actually a decrease. So this is greening followed by browning, if you will. We're calling, just for later in the talk, when the trends give this qualitative pattern of an increase followed by a decrease, we're just calling this a hat-shaped trend. So the next more tricky part is we've done the detection part of it. Now we want to do the calibration part. So we need to calibrate a process-based model that is capable of reproducing the NDVI data that we're observing, or the ecosystem dynamics that we're interested in. And so I've been using this little model in a variety of applications. This model is capable of simulating plant growth as forced by a bunch of environmental variables, in this case air temperature, photosynthetically active radiation, atmospheric CO2, moisture levels in the soil, nitrogen levels in the soil, and also the temperature of the soil. So you'll notice there's some things missing in there. But anyhow, we then implement this in what is called a state space modeling approach. It's also called the hidden state modeling. Essentially what one does is one models two time series. The first time series is, if you will, is the hidden state time series. So this is there's some hidden process which is generating the data that we're observing. That's the second time series of NDVI. So we use the process-based model to simulate through time how the system's dynamics as forced by the environmental forcing variables. And then we use the state variables in that model to then predict the NDVI, the observed system states at given points in time. We use Monte Carlo Markov chain methods to then fit these models. This has a bunch of advantages because you can separate your observation error from your process error, and you can also get reasonable or coherent estimates of your uncertainty in your model predictions. So here I've got two examples of two different sites where the model is fit to the time series. The top one is the time series you've already seen. It's a pretty regular time series where there doesn't appear to be much interannual variation. And the second one is where we actually do have a little bit more interannual variation. So this is just to show you that the model, at least qualitatively, seems to be doing a reasonable job of reproducing the data. Now on to the attribution step. What we're seeing on the graph on the left there is the ability of the model to predict the observed NDVI anomalies. So we're looking at the modeled NDVI anomalies and the observed NDVI anomalies. The red line is showing what the full model does. And I mean, there's a lot of noise on that, but essentially it's in this case pretty close to the one-to-one line, showing that statistically the model is able to describe the trends in the anomalies. The blue line is showing what happens when we remove the trends from the forcing data that is used for the model. And then we get something which is not too, or a slope which is not too different from zero, showing that when we take those anomalies in the forcing variables away, that we can't reproduce the anomaly, suggesting that that some of those trends in the forcing data are responsible for the anomalies. The right-hand panel is kind of showing the sensitivity analysis, where we try and isolate which one of those environmental drivers or which sets of anomalies in those environmental drivers is responsible for producing that pattern that we are observing. In this case, so the candidate things are air temperature, temperature of the soil, soil moisture, solar radiation, or PAR, and then the atmospheric CO2 concentration. And in this case, and the spoiler is that this is actually most often the case, is that it's soil moisture that is explaining those patterns. So that's the methodological overview. Let's go into the actual study. So we're using this GIMS data set. This is a huge pixel size, and like Edward mentioned, in our lab we petrified of confounding land use change effects from ecosystem dynamic effects. So we wanted to find wilderness pixels, large pixels where the vegetation is relatively homogeneous and there's no evidence of land use change during the time series. So that kind of restricts us somewhat. For my Brazilian friends, either we didn't look hard enough, or you guys don't have homogeneous vegetation, or you have a lot of land use impacts. So on the detection side of things, what would we find? That hat-shaped trend that I described earlier is the most common thing that we see in the data, and this may or may not worry you depending on your ideology, but essentially it's showing that we're having initially an increase we're seeing greening trends followed by browning trends, and this essentially, there's some geographic pattern in how this is playing out, but it's pretty widespread. On the attribution side, as I alluded to, we're essentially finding, when we look at the Savanna sites we investigated, it's always soil moisture that is explaining the pattern, or explaining the ability of the model to explain the anomalies. So I'm on to my conclusions already. What we found is that using only climate system drivers, we can explain NDVI anomalies and attribute these anomalies to the drivers. Surprising for me personally, and I think for the other authors as well, was that the CO2 effects were weak. When we started the study, we thought we'll be able to show the role of CO2. And essentially afterwards, and it was also tricky convincing the reviewers of this, but I think that we can explain it by saying, well, if you're a plant, you can't benefit from elevated CO2, from a CO2-enriched atmosphere, if you've got your stomata closed. And I think that this is perhaps what is happening in this data set. The caveats are many. I could make a very, very long list, but I want to highlight a few. We're using big pixels. This lumps grasses and trees together into a green slush, and this is obviously suboptimal. The other is that NDVI is an estimate of vegetation greenness, and this is not necessarily always well correlated with light use efficiency and GPP. Sometimes it is, but often it's not. So going back to our CO2 conundrum, if the spoils of CO2 fertilization are not used to produce more leaves, well then the satellites measuring NDVI are just not going to know about those CO2 fertilization effects. So that could be one of the reasons why we're not detecting the CO2 fertilization effects that we might have been expecting. So for more details, you can check out our paper on this, which came out a few weeks ago. And thanks for your attention, and hopefully some time for some questions.
Thanks, Steve. Hot off the press. So we've got time for a question. Sandra?
Hi, thanks, Steve. I was wondering why you chose soil moisture over rainfall.
Because plants don't have rain gauges, and they take up their moisture from the soil.
The reason I ask is we did some local scale modeling of rainfall, and we looked at the patterns that were produced by a locally tweaked rainfall model that gave us a 1 kilometer resolution rainfall data set monthly from 1981 until 2018. And the patterns look really, really similar. So I'd be interested to see how they compare if we just overlaid some of that results.
On the soil moisture data that we used.
Yeah.
Because I'm like squeezed for time, I didn't mention that all the environmental forcing data that we're using comes from the Area 5 land product. So this is a reanalysis product which has a whole bunch of climate system variables, which a process-based model simulates this, but in a way that it's consistent with available station data.
Thank you.
Yeah.
How are we doing for time? Sorry? 54 seconds. Who's got a 10-second question and a 30-second?
Maggie had her hand up right in the beginning, but
Question for clarification. I see you were desperate to ask a question.
Hi. I was just interested, what this elevated CO2, the greening of terrestrial biospheres that we're kind of predicting, but we can only observe it above ground. What if all of that extra productivity goes into the soil, into roots? How does that affect your projection, your forecast then?
well, that's the caveat number 2, right?
Yeah.
It's a problem. I mean, we need to find out what's going on below ground, so it's a challenge that we need to expand our observation window.
Thanks,
Okay.
Thanks, Steve. Great presentation. Going to move on to Mulalu Savanna, and she's going to look at climate and disturbances in predicting some of these patterns. Thanks, Mulalu.
My name is Mulalu Savanna. I'm a PhD student under the supervision of Thomas Hitler and Matthew Forrest. I'll be presenting to you a part of my PhD study, which is, I've changed a little bit my title. It's the role of climate and disturbances in shaping South African grassland biome. Earlier on in the week, there was a presentation talking about something about there's almost nothing new under this tropical sun. And this is something which is true, because we already know that climate and disturbances contribute to vegetation distribution. But what does this mean when it comes to vegetation modeling, specifically modeling grassland in South Africa? As you have already figured out, I'll be looking at South African grassland and with its neighboring biomes, Savanna and Namakuru. I will not go into details on what actually constitutes the different biomes, since most of us know about that. So what we did for this study is that we tried to investigate the defining limits of the grassland and to see whether indeed these limits do apply, and if they do, can they be explained by just climate alone, climate data. So what we did is we tested 3 climatic factors and 1 disturbance-based factor. We used Area 5 data for the first 2 factors, which is a 0.1 degree resolution, and for the other 2 factors we use CRU-JR, which is a 0.5 resolution. The reason for this is because this is a continuing research, where we started by just using only a 0.5 resolution, and now we are gradually going to a 0.1 resolution for a better analysis. So the first factor that we looked into was the minimum temperature, where according to Prentice et al., tropical trees cannot grow beyond freezing temperatures of a monthly minimum 15.5 degrees Celsius. This is a hypothesis which is being used by most global models, and it's good to question whether this is still relevant when we look into regional scale. And also when we look into recent data that we have, which is available, which is a daily data, does this have any difference in us addressing this factor? Also, there are various research which has been done and expressed that plants do not really die from just being exposed to cold. So there are other factors that actually contribute to trees dying. So what we did was to test both monthly and daily minimum temperatures for this factor. We further tested a water budget factor where it's already known that lack of soil moisture reduces tree and grass competition. And according to Detroit, Namakaru grassland boundary is determined partly by rainfall amount. And according to Masubelele, reedification is predicted to contribute in Namakaru shifting. So with this, we tested the water deficit, which is the difference between precipitation and potential evapotranspiration. The other factor that we tested was the growing degree days, where according to Woodward et al., heat sum of the growing season controls the distribution of grassland biome. So what the question that arises is there enough heat accumulation for woody vegetation to overcome the local disturbance regime? So we tested this factor. And the last factor that we tested, which is a disturbance factor, is with regards to fire. Yesterday we had so many talks talking about how important fire is for vegetation distribution, and there were so many research which has been done confirming this. And since we already know this, the question now arises is how much fire is thus needed for tree mortality? So we decided to prescribe fire to attest for this. With the first results that we got is that, as you can see from the map, we got a minimum monthly temperature limiting from the north and east of 0 degrees Celsius. When compared to daily minimum temperature, we found that the limit is -2. And this is very interesting that both the two limits are different. We also recognize there is a different factor that is actually limiting the western side of the grassland and the Namakaru, on the Savanna, where we find that the two differences is most likely because there are two types of savanna in play. And we have realized that global models do not account for these two types. Looking at the histograms, we found that savanna do not grow below -4 degrees Celsius, and this is for minimum monthly temperature. And for And for daily minimum temperature, we found that the savanna doesn't grow below -6 degrees Celsius. And this is a 2-degree difference, which is something that maybe we could look into going forward. Of course, we saw that the grassland and Namakaru didn't have a clear separation with regards to this factor, and meaning other factors are actually contributing to that boundary. Looking at water deficit, we did see a clear separation of the grassland Namakaru boundary, and we found that the Namakaru boundary doesn't grow above the water deficit 1,500 millimeters, and this is where the water availability can be kept. And with the savanna and grassland, we didn't find any clear separation. So this leads us to the final disturbance factor, where what we did was to simulate standard LPG gas. This is a a global vegetation model, which runs on a SimFire-based fire model. And then we further conducted simulations using Spitfire model. The two fire models didn't represent the fire well. That is when we decided to prescribe fire. This is, we conducted simulations using the same Spitfire, but this time around we prescribed it. We prescribed it from 1 to 20-year return intervals. And this was because we wanted to investigate how carbon mass relates to the return intervals. And what we found was that a gradual increase in tree coverage as fire return interval increases. And then we found that there is higher amounts of grasses in return intervals 3, 4, and 5. We found that the grasses peak at 5-year fire return interval, while tree saturates at 9-year return intervals. So basically, we have gone and investigated the different factors, and the final thoughts that we have from this is that indeed climate can explain minimum temperature along the savanna and grassland biome. However, it is important to realize that the monthly and daily temperatures have different results, and this is a 2 degree Celsius difference. Different factors are contributing to the western side of the grassland and the Namakaru, and this is because of the two defining types of the Namakaru, which the global models do not account for at the moment. Maybe it's something that to look forward to when modeling in the future. Climate can explain the moisture deficit factor, and this is only in the grassland and the Makaru. And the water deficit can be kept at 1,500 millimeters. So prescribing fire of 3 to 5 fire return interval could be advised to better simulate the South African grassland. However, this can be taken with a grain of salt because when I did this was covering the whole of South Africa, not really zooming in on just grassland. Climate, so we can conclude that climate indeed is important, but alone it falls short. It's important to include fire when modeling vegetation. Going forward, my future steps is that, as I mentioned before, this is an ongoing research, and at the moment I'm currently simulating the model at a higher resolution of 0.1, and this is for better analysis and also trying to see how the model will look like when all these parameters, all these factors are applied. I also would like to zoom in into just grassland and see how the carbon mass relates to return intervals. I would like to thank all my collaborators, and thank you for listening.
Thanks, Mulalu, for sticking very well to the time. We've got 3 minutes, so if there's some questions. Kevin?
Really interesting talk. I'm just curious, do you have any plans to modify the base model to represent grasses in a different way than LPGA Guess does currently?
Yeah, that is the plan. Because initially, our initial results were showing how bad the model was. So that is why it led us to trying to figure out exactly what are the different factors that are contributing to defining the different boundaries. And the hardest one was actually not including the shrubs one, the fine boss one, but the hardest one was the grassland. So it was giving us so much trouble, so we had to sort of like dig deep in what is actually happening there. Yeah.
Thanks so much, Mulalu. We're going to move on to Lucy Wells, who's going to take us to Latin America, the dry tropical ecosystems.
Okay, so hello, my name is Lucy. I'm going to share with you the results from the first chapter of my PhD, which is looking at vegetation structure in Latin America. So accurate biome delineation is important for lots of reasons, such as deciding on appropriate management and conservation planning, and modelling responses of vegetation to global change. But tropical dry biomes, such as dry forest and savanna, are quite hard to delimit based on traditional methods of using climate descriptions because they occupy similar climatic space. So this makes it quite hard to answer these questions about management and global change. So my project is looking at vegetation structure as an alternative way of delimiting these biomes using remote sensing data. So I've done this initially using the case study of Northeast Brazil. So you can see my study region up here. So this is part of the Brazilian dry diagonal. It's mostly made up of the Caatinga, which is dry forest, and sort of quite a mixture of wet forest and cerrado, which is savanna in there as well. So to do this, I used 7 vegetation structural parameters to describe the structure. And these were measured using remote sensing. So I've got biomass, and biomass error is kind of a measure of heterogeneity in each pixel, the seasonality, and the presence of fire as a proxy for grass, and presence of grass, and looking at LAI and canopy height as well. So I first ran principal component analysis over these to see which are kind of most important. Sort of vegetation abundance is the key driver, which is a mixture of biomass and LAI and height. And also fire and seasonality come out as important. And then I clustered over these PCA components using a number of different methods. I'm going to share the results of one of those. So it comes out with this map. So this identifies 6 vegetation groups in this region. And I've also got the box plots of the values of each structural parameter in each cluster. And I've used these box plots to try and describe and understand each structural group. So just to highlight the ones that are quite easy to identify, the ones highlighted in yellow, so that's wet forest. So it's got the much higher biomass, it's not very seasonal, and taller trees. And also the orange group, which is the cerrado, which is easy to identify as the one that burns the most. And the other ones that I've kind of tried to describe and identify is the blue, so that's the transition, I think, between cerrado and caatinga, and the others are sort of different forms of dry forest. So just a couple of photos to try and illustrate to you what I mean by these biomes. So the top one, we've got the cerrado, so you can see it's much more grassy. In the middle, sort of the classic caatinga, so you can probably spot some patches of bare ground and some cacti in there. So that's the turquoise group in the map. And at the bottom, more arboreal caatinga, which is functionally similar but has much taller trees. And then to try and understand what's driving these clusters, I've done some random forest regressions. So you can kind of see that I've grouped these into different types, and the red ones come out as most important. So that's telling us that different soil variables are driving these clusters. And there's only one climatic variable, which is precipitation in there. So that's linking back to what I was saying at the beginning, that climate isn't that good at describing these different structural groups. There's also some human factors in there, which I was a bit surprised at, and I'm still figuring that bit out. Yeah, so just to conclude, vegetation structure does kind of work to identify different biomes in northeast Brazil. This is likely driven by soil and human factors rather than climate. And my next steps are going to be comparing this to other methods of mapping, for example, looking at floristic maps. And then I'm going to be shifting my focus to looking at southern African dry biomes to see if this works there. Yeah, thank you for listening.
Thanks, Lucy, for sticking perfectly to your time and for a brilliant 5-minute talk. Thanks so much. Next, we've got Graham von Maltitz, who's going to talk about using satellite imagery to look at degradation in savanna systems.
So mine's a joint presentation with myself and then from a group of people from Jena University in Germany, and particularly Marco, who's helped me pull together a number of the time series in here. And really, I'm just going to be looking at sort of some exploratory look at data rather than giving really hard analysis or conclusions. But just to start off, why do we need to monitor degradation? And I think there's a whole number of reasons, and I've just mentioned a few of them here. And the first one might be a rather strange reason for people sitting in an audience like this, but we do monitoring for international reporting, and unfortunately for us, this drives a lot of behavior. What goes to the conventions gets forced into policy, that comes back and has implications to the country. We also need to understand where to implement projects. And here I'm thinking more off the reserve in the sort of areas outside of reserve areas, particularly identifying where restoration should take place. And this has been a major issue in the country for a long time. We've got different funding streams coming in for restoration. Where should we focus the effort? And one of the things that I'm involved in a lot now is, for instance, ecosystem-based adaptation. I really believe ecosystem-based adaptation makes sense where it's a response to an actual degradation. Then we can get sort of win-win benefits. If we start doing adaptation things or mitigation projects outside of degradation, then we get these sort of problems that were mentioned the other day, putting trees into grasslands, etc. But then the next side of that is, once we've decided where to do a project, how do we know we've actually had a positive impact? How do we know that what we're doing on the ground actually is making a difference? I think another aspect that's coming up that is very interesting, we also know that most of the biodiversity actually sits off the conservation estate. It's out there, particularly in the rangelands out there. We need to understand what happens to the biodiversity out there as those areas degrade. We also need to understand how good those areas are from a conservation perspective, from a biodiversity perspective, and more so from a climate change perspective. How good are those areas for allowing species to move through in a changing environment? And then this is all confused and conflated by the fact that we've got climate change impacting as well as the human changes at the same time. I think at the moment we've got one of those change points in history when it comes to remote sensing. We've got a whole lot of old data that was really expensive. If those of you that are my age will remember the first days, we still got Landsat data. You had to save up on projects to buy just one scene. Now that's all becoming available, and I'm going to look a lot at the Landsat data, and particularly the work that Fenter et al. Did in making this data available and analyzing it for South Africa. We've also got a whole lot of new satellites that are going up there, also open access data, and we're going to talk a bit about some of the data coming out of the Sentinel-1 and 2. And Chris later on is going to do that in a lot more detail. And then we've also got these new processing facilities. We can actually process data in ways that we never could in the past, at speeds we never could in the past. But we have this dilemma, and particularly when we start looking at national maps of degradation, we actually haven't a clue what's going on there. And I've put up basically 4 maps there that are coming off using a tool set, Trends Africa, that's used by the Degradation Convention, and it gives the different options of how to analyze where degradation is taking place. As you can see from that, you could literally choose any point in the country and either get it to be a positive or a negative, just depending on how you manipulate the input. And just for comparison there, I've got the Landsat data from Venter, which we'll talk about later. So what are some of the main degradation challenges? And I'll talk in sort of also mapping challenges within South Africa. And the one is defining the baseline. And this could be an absolute talk, a whole conference, in fact, on its own. You know, when we say something's degraded against what? And there are different ways of looking at it. We can look historically. And is it before white settled in South Africa, before black settled in South Africa, before the Khoisan settled in South Africa? You know, we can choose any aspect we can think of. We could also say what we do it against what we think the vegetation should like. I mean, we're all ecologists in the room. I mean, surely between us we can say exactly what a savanna should look like. That should be really easy. Okay, there's a few chuckles there. But in a sense, that's what we did in the old days. I think in a sense that was how lots of parks were managed. We can also do it against set times. The LDN, the Land Degradation Neutrality, uses 2015. The Climate Change Convention tends to use the start of the Industrial Revolution. Then the other thing that we have is a huge problem in South Africa is, and I think they are two biggest degradation problems, it's alien invasive plants and bush encroachment. And the reason I really highlight this is, one is they probably cover more of the country in terms of degradation problems than anything else. But the other is that they give what we'd call a false positive on most of the measures. So you look at an NDVI trend, a vegetation index trend, and the vegetation looks like it's getting really better and better. And this is what we're saying to the rest of the world. Look, the world's getting really better in South Africa, but it's all because of aliens, or it's all because of bush encroachment. Then we have other types of rangeland degradation, but particularly, say, for a farmer, species composition changes. It changes the carrying capacity, but it might not come up at all on a mapped product. And then we've got, as I think it's come up so many times already in the talks, the fact that most of our systems here, particularly the savanna systems, are degradation driven. So we've got fire impacts. How long were you since the last fire? If you go into the fynbos, you might have a 20-year fire return time. Are we just tracking recovery from fires and then seeing the effect of a new fire? Similar problems in the savanna, but slightly less return times. This huge interseasonal variability in rain, you know, runs of maybe up to 10 years of really dry years, runs of very wet years. How do we account for that in our mapping process, particularly when we've got very short time series of data? Also the fact that we've got 9 different biomes, each one of these has very different processes. Understanding the different processes in the biomes is very important, but it's going to lead to different data. And then we've got to decouple the climate change from the human-driven aspects of degradation. So going on to the Landsat EVI data, it's a very exciting data set that's come out. It's a long time series going right back to the late 1980s, up to more or less, I've got to miss, up to, in the case of this analysis, the 2018. But it's a very patchy data set. Some years is a lot of data, some years is very little data. Some years you might find there's 6 data records in December when we've got some of our most growth. Another year there might be no data records in December. So you've got to work, it's a mucky data set, but it probably is fairly good for trends, and probably good for trends when you in an area very close to each other. Shall I just keep on talking? Is that loud enough for? Okay. I've got to find my buttons here. Okay, it's going to. Let me.
We can wait for the power.
Let's wait for power to come back, because it's going into images that are not going to make sense without the without the.
Questions at this stage, if you're happy to take questions.
I'm sure, yeah.
Sally?
No, I just wanted to make a comment. I don't know if you know the project in Brazil called Map Biomas.
Sorry?
Do you know the project in Brazil called Map Biomas?
No.
So this is a project mapping the entire country about vegetation types, but they are also mapping land use changes, and now they have this project about mapping land degradation from 85 to 2021. So maybe it would be good to have a chat with them as well, how they're doing and so on. So just take a look at mapbiomas.org and you can find everything, how they're doing the methods, and it could be like helping also.
Thanks.
Sally?
Yeah, thanks. I suppose I just wanted to follow up on your comment around how to define degradation. And everyone laughed when you said how we think of vegetation should look. But in a way, I think if we all agree that we are totally biased when we try to think about degradation, it might be a productive way going forwards. Like, to me, that is the best way to define it. It's like you all decide, what do you think it should look like? And then you can quantify whether it's degraded or not. So I suppose you know more about the global degradation. Is anyone pushing that sort of definition, or are there still just sort of sensitive trends in India?
Yeah, I mean, the one thing at the moment that we've got is we've got this whole program called Land Degradation Neutrality, and that's got 3 indicators that are used in that, and the one indicator is trends in NDVI or productivity. So at a global scale, productivity is being seen as a major indicator of degradation. The other indicator that I think is very useful is soil carbon, and I think we'd all love to see soil carbon used as an indicator. Unfortunately, the only way we measure soil carbon at the moment is through changes in vegetation cover. So you've sort of got this circular argument. You know, you think you've got a nice independent variable, but it's actually no different from your productivity variable.
So you don't think like a values-based approach, which has nothing to do with biophysical measures, is used?
That has been used. If you look at the work that Tim Hoffman did almost 2 decades ago now, that was very much a value-based approach, getting a consensus approach per biome. But what that doesn't let you do is it doesn't let you get a very strong spatial understanding of what's happening, and it's very difficult to monitor change against that in a rigorous way. I think you probably could do it, but it's slightly problematic. Are we going to get power back again?
Yeah, it will come back at some stage. It will be quite clear when it's
We'll notice it.
Is that a follow-up or?
It's a follow-up, but someone else has a question.
Okay, Tony? Okay. Let me wait till then. Okay. You're perfectly halfway into your presentation.
Perfectly,
so
okay.
Stop the clock.
Super.
And we'll just wait for the...
Okay, well, while that's coming up, so when I saw the set of data, I thought, I'll do what everybody does with a nice new set of data. Go to somewhere I know well and see what the data does in that area where we know well. And I was fortunate. I grew up with family having a farm on the Magaliesberg. I've thought there's a bit of degradation taking place there and a bit of bush encroachment and so on. So I thought, let me home in on the Magaliesberg range, on a property I know, on a property I've been visiting for way longer than this data series exists. So this is the area in the Magaliesberg. Put Fenter's EVI data onto it, and you get this striking pattern coming out there. The entire Magaliesberg range from one end to the other is a sort of picks up very clearly there. And firstly, one thinks, well, okay, that's degradation. When you actually look at it, it's not really, some of it's degradation, but a lot of it, that sort of lemony color is actually more or less stable. So, in a sense, that's what you want to think. Here's an area that's got almost no land use on it. It's very poor quality for grazing or anything. It's actually a national protected area nowadays. And maybe that's what you expect. On a long-term trend in EVI, if you're in a protected area, things should actually stay the same. There shouldn't be reason for trend one way or the other. But then you have to ask, why is the valley to either side of it greening up so much? And if you look at that, you can pull out some areas. For instance, there's the platinum mines. They're clearly, big red spots on it, some fields and so on. But the rest of it's a muck of different land uses and is almost all greening up. This is just looking at that sort of Magaliesberg area that's not greening against long-term rainfall. And interestingly, very, very similar patterns, almost identical patterns, neither of them statistically significant, but showing there might be a bit of link between what's happening there and the rainfall. And I think what we're seeing here is off the mountain. Okay, so the Magaliesberg, for those of you who don't know it, it's ancient quartzite. It's very, very nutrient poor, very leached soils that it produces. Off the Magaliesberg, you lean onto fairly heavy clays. So I think what you're picking up here is off the Magaliesberg, you've seen a trend of CO2 impact on the vegetation. You've seen an overall slightly greening up due to CO2 fertilization. On the Magaliesberg, I think what's happening is that soils are so poor that you're not actually getting a CO2 compensation effect taking place. And there is some literature out there. Fortunately, I did a very quick Google. I haven't looked into this in any detail, but it seems there is possibly some link to this, that where there's low nutrients, you don't see a CO2 effect. I then thought, okay, let's move to Kruger, take some natural experiments here, and see if we can pick up similar ones. So the first place we looked at is what we call the Rhone camp in northern Kruger. Area fenced off, most of it since 67, some of it since 80. Very big difference visually between inside and outside the Rhone camp. When you look at the EDVR, almost no difference at all. If it wasn't for the fire breaks around the area, you probably wouldn't pick it up at all, except for the side. Think about it, it probably makes sense. Both areas have sort of stabilized, and you've just seen similar trends, but with a different base. And so pulled out the data to see if that's the case. There is a little bit of evidence that that might be the case inside the Rhone camp, slightly higher NDVI than outside. But in truth, very, very little difference. And the difference between inside and outside the camp over the long-term means is probably a fraction of the difference you see in between-year variation. So, between-year variation just absolutely overruling those slight differences in trends. I decided to look at areas where there's a far greater difference inside and outside of Kruger, choosing an area. Let me show you the area there. So it's where some of the long-term burning plots on your Pretoriuscorp. You do see there a similar idea. Outside of Kruger, a sort of lower base that you're starting from, but fairly similar trends. And here we tried to then pull in some extra data, and we haven't analyzed this yet, but just to show you the new data that's coming available through the Sentinel platform, how you can use this set of different data and actually start picking up a lot of really interesting patterns taking place there, similarities between all these different data sources. And again, that will be picked up a bit later, to talks from here. Tried a similar thing in the Skukuza long-term experiment with an exclosure. Some areas, no elephants, in fact, almost no grazers at all, and no fire. Other areas with fire and no fire and elephants. You see a big difference on the EDVR. Interesting here, you see almost no difference between the patterns until the exclosure was put in place. So I think what you're seeing here is it's a new exclosure, new dynamics taking place. You've seen a splitting out of the data since then. I think a lot of interesting things there. I try to separate out the sodic and non-sodic areas. Probably a lot of stories there, but very small sample sizes. One probably needs to be a bit more cautious about it. But certainly sort of type of patterns one's expecting. Again, you can take the new data, run, for instance, radar backscatter, seen very different seasonal patterns coming out there. Huge differentiation in some times of the season. Similar with NDVI. And then so to move on to some conclusions from this. I think the first conclusion is that we actually now have more data than we've ever had before. But we're really at a point where we've got the data, but we haven't yet figured out how to fully use it. So it's sitting there waiting for clever people to figure out how we can use this better. But also that you can get very similar signals, particularly when you're looking at long-term trends that mean very, very different things. So any product that simply looks at a long-term trend and thinks it's giving you a useful endpoint is not very, very helpful at all. And this gets, when you make sort of broad assumptions, particularly when you start doing this across the country, it gets really problematic. So we pick up bush encroachment, alien species, croplands, all sorts of things as restoration, and probably CO2 effects, all looking like good restoration across the country, probably not actually be what we as ecologists would want to think about. So site-specific interpretations are often needed. When you look at this data in a site-specific way, it often makes perfect sense, but you can't really extrapolate beyond it. Not all results are what we'd expect, but normally we can understand, we start digging in, understand what's going on. Trends don't tell us about the state of the vegetation, and we make that mistake often, that we look at trends and we think it's equivalent to state. And I think we can maybe learn a lot from the fluctuations, different aspects. I think we had that in the first talks today, taking out different components of the data, not just a straight, simple trend. But I think where the exciting part's going to come is trying to mix different data sources. We've got this multitude of different data sources. How do we mix and match to get out new patterns, new things that we didn't have in the past? And I think that's where we're going to make huge gains in the future. And I'm being told that my time's out, so I'll stop exactly on that. Thank you.
Okay. Thanks, Graham. I think you've had ample time for questions. I think in the interest of time, we're going to move on. I just want to tell you about a poster by Hilma Nkihalwa, that's from Jena University. So please, that's looking at mixed pixel analysis of sentinel data. So have a look this afternoon, please. And then we're going to move on to Tulia, and she's going to give us a talk on the newest woody cover map, but the oldest one at the same time. So she can explain that.
Thank you. Okay, so good morning. I am Tulia Riccardi and I'm a PhD student from the University of Rome La Sapienza. And today I'm going to be talking to you about part of my PhD work on creating the first woody cover vegetation map of Kruger using historical aerial photography from 1939 to 1944. So the dataset that we use is only part of a bigger aerial photography dataset that comprises images from the 1960s, the late 1970s, and then from 2008 to 2018, every 2 years. So our first objective was to create a historical baseline of what the woody cover was like in the early years of the park's designation. So as you can see here on the image on the right, our images do not fully cover the park area, so it's about 85% of that park, so still a very large area, so 1.7 million hectares. And there were two main challenges for this study. So the first one was how to create a methodology to classify woody cover from black and white images. And the second one, as you can see on the right, it is a composite of images from different years. So how to do that on a large scale. So to address the first issue, we decided to use object-based image analysis because our images are in black and white, so it's a grayscale. So we couldn't use a traditional pixel-based classification. Instead, we use this object-based image classification. So what it does is that it groups pixels together into these vector objects, and this process is called segmentation. So once we have segmented the image, we create our training and test sample based on what kind of land cover classes we are interested in. So for us, it was woody and non-woody cover. So after that, with machine learning, and we use random forest, we used this training and test samples to extrapolate and classify the entire aerial photograph. So we decided to look into different softwares. So in the beginning, we started with more technical softwares like eCognition, very expensive. But then in the end, we decided to go and use Google Earth Engine for several reasons. So one of the most important reason was that seeing as how we were working on such a large scale, we needed to have a software that was very easily usable from different users, especially to do the training set. And then it's also open source. And then we really wanted to do something and have an algorithm that is iterative and an iterative process and replicable. So once we had the tool sort of figured out, we had to pass to the sampling strategy. So how were we going to create our training and test samples? So in the beginning, we thought, okay, let's do a random generated points across the whole aerial photograph. But as you can imagine and think that if we're in a savanna landscape, the probability of a random point falling onto a woody cover was low. Therefore, what we had in our first results, that the algorithm was classifying really well the non-woody cover, which we were not interested in, and not the woody cover. So what we did is that we decided to add another geometry that we called add trees, as you can see here in red. And so what it did is that it helped the algorithm classify the image for the woody cover. So we gave it good examples of what woody cover was. And then it also helped the random forest because we had the same number of training points per each land cover for each land cover class, and this helps the algorithm also classify better. So, and we sort of called this approach the random convenient object-oriented approach. So we first tested this algorithm on smaller areas, which are not that small because they're 25 kilometers squared. And we call them subplots. So as you can see here on the image on the bottom left, we have the subplots in red. And we placed them in 5 vegetation types in the southern part of Kruger. And so we had our results on the right. So these are our initial results. We had actually good accuracy measurements. However, we were interested in scaling up the classification. So we decided to, again, use the vegetation type as our unit. So we had 2 ideas. So one was that, okay, so if we obtained a good classification with good training points in the subplot 1, as you can see on the bottom left, we could just scale and use those training points to scale it up to the entire vegetation type because it was representative of the same type of vegetation. But then we had a second idea, thinking, okay, instead of one big, large subplot where we do our training, we would have several small areas across the vegetation type and do our training in those. So we actually had good results for both of them and actually comparable. But our main objective was, again, to scale it up even further. To the entire Kruger National Park. So now I come to our second main challenge was, okay, so we have a composite of images from different years. So how do we address that? So what we decided to do is that we worked at the photo job level. So photo job is photos from the same year. So aerial photographs from the same year. But we still wanted to maintain the idea of the vegetation types. So what we did was that every photo job, so called the aerial photography, we divided into smaller areas because they're still very big. And then what we did, as you can see here on the image on the right, we took the total area of that study site. Then we took 5% of that area. And then we counted how many vegetation types were present inside that study site, and we placed subplots within every vegetation type. So we went back to our second alternative idea of having several small subplot training points. And so in the end, our final classification was obtained per photo job, so per year. So this is a summary of our results. So what we see actually is that in the northern part of Kruger, so in the 165A on the top, and then in the southern part, so 155 and 150, we actually had the less amount of woody cover, so about between 21 and 23%. While instead the majority of the woody cover is larger in the middle part, so 165B and 56P. So just so I don't only talk about numbers, I wanted to show you a little bit about, in depth, our sort of zoomed in area of our woody cover classification. So this is a small part, so about 0.5 kilometers squared inside just close to Punta Maria. So as you can see here, the bigger trees, so the bigger woody cover around the river is highly visible and it classified really well. This is where also it becomes stronger, where stronger the object-based classification. So as you can see, the object of the river is well classified, but it still picks up a lot of the woody cover, the smaller woody cover around the river. And that is it was in the granite part, geology. So this is instead close to Satara, so south of Satara. As you can see here, it's in the basalt area, so I chose sort of just to show you a difference. And as you can see here, there's a lot of so that you can really see the woody cover, and it picks up a lot, and it has a lot of contrast between the woody and unwoody. You also have to remember that these images are taken during the dry period. And then this, I also wanted to show sort of an image of the Malelane Mountains. So as you can see here, again, you see the river and the rocks in the north, the top part of the image. And again, there's a lot of contrast between the woody and the non-woody. So we also calculated the total woody cover of the composite of images. So we have about 438,000 hectares of woody cover. So this is based on the 1.7 million hectares that we have for our images. So that's about 26% of the park. So obviously, once we have our map of the historical woody cover vegetation, what comes next? So what is most interesting is obviously the analyses and that we want to do. But unfortunately, we didn't manage to get to that part yet. But what we want to do next is to extract the historical woody vegetation for geological, pedological data sets. So as you can see here on the right, this is a basic geology map between basalt and granite. See if we have a difference in the woody cover for bioclimatic gradient. And then also if we have any management units to see if there was any sort of difference there. And then I've been talking to a lot of you about this work, and everybody's always interested. Oh, what about the change? And obviously that's the most interesting part is, what has changed since back then. So once we've created this huge historical data set, how is it different? So we actually are finalizing the 2012 images, so the aerial photography. So we decided for 2012 because it was prior the drought of 2015. And so we're going to look at the changes in woody cover between the 40s and 2012. But then we're also interested in the temporal spatial analysis of the other data sets. So the mid-60s, the late 70s, and the 2000s. Mainly to assess how the woody cover has changed based on different management strategies. Things have changed so much since the 40s to now. So that's what we really want to move towards, is also the analysis of change. Here I wanted to show you just a sneak peek of the 2012. This is again in the, so I can just go back and forth. It's again in the northern part of Kruger, so just again around Punta Maria. So just, this is interesting, at least for me. So then just one last thing. So we really wanted to go back into my first sort of slide, is to make this methodology iterative and replicable. So aerial photography of Kruger is periodically flown across. And so what we really wanted to do with this methodology is create a monitoring system of the woody cover. So what I did here is that I took the boundary of Kruger, huge, and I divided it up into 31 study areas, each about 620 kilometers squared. And then I also, as I mentioned in the methodology, created a layer of training subplots, again taking the 5% of each study area and then putting every study training plot within each vegetation type. Especially in the north, as you can see, there are lots of really small vegetation types that cross the study sites, and if it was really, really small and our subplot couldn't fit, we put the biggest area that we could, and then we took the rest of the area and we put the subplot in the closest, biggest vegetation type. So that's just very technical. And so once you have these two layers, and then you can upload the new aerial photography, the layer of the training areas and the study sites here in black, and then you can create, and then you just have to create the training and test samples for every study site, and then you can actually export the object-based classification and obtain a woody and unwoody cover periodically every 2 years or when you have it. So really assessing and monitoring changes in woody cover. So I really want to thank our students from the master's course from Professor Attore that really helped us in creating this training and test data set. They were actually really sad that we were not live streaming today because they were all ready at 8 o'clock in Italy. Anyway, so they really, really helped us, and without them it wouldn't have been possible. And then all of my co-authors and collaborators that helped me implement and create this methodology. Thank you.
Thanks so much, and I think we look forward to all the next iterations as well. And thanks for translating it into a monitoring tool that can be used in future as well. We've got just one question. Time for one question.
Thanks for the wonderful presentation. I just want to know, if you were to add more classes, will it reduce your overall accuracy? And another one, did you look at the confused error matrix in terms of the classification? Thank you.
Yes, so we did. I didn't include it, or else I would have too much. Didn't fit on my slide. So if you're thinking about the classes, I mean, unfortunately, we only had two classes, so woody and non-woody. But for the black and white images, it was already really hard to decipher what was, the woody and the non-woody. So unfortunately for that, we had to have just two classes. So obviously, even all of the matrices are just, two classes. So obviously, that's difficult. But going ahead, in the 2012, we used the same methodology to make it comparable. But definitely having the newer images that are not in black and white, we could definitely add more classes. And so obviously just to do the change between the two would definitely, I think, increase the accuracy and also just even have other layers and not just non-woody, which is, I don't know, basic, but black and white is scales of gray. I think now I see in black and white, I think in past year.
Thanks so much. Next we've got Chris Schmulias, which I think was very close to beating Greg to having been at the most network meetings. So you must share some of that chocolates with Chris.
I wonder, I think 14 maybe. Jussi and I are always surprised how often we were here, but it's a regular thing. So, well, thank you very much for the opportunity to give this presentation. Long title. I'm presenting some results of our SunParks project called Cold EMS, which is a mixture of the Spaces projects EMS Africa and SALDI, which you can see down here, the logo. The cursor. Okay. SALDI stands for South African Land Degradation Indicator. So you may wonder what it has to do with SunParks. We have 6 sites all over Africa, each 100 by 100 kilometers. And in 4 of these sites, there are obviously national parks. So we have Agulhas National Park, Ochabis National Park, we have Kruger National Park, and Mokala. And so we have chosen these sites also to have always to be able to map inside and outside protected areas. Then the focus was what kind of land degradation indicators we should actually focus on. And that came from a publication by Graham from Maltitz. No, can I just go? Okay. And I have, of course, a Mlik mouse. Okay, so here you see the list of indicators that Graham also just has shown in his presentation. But thinking about remote sensing, what is it what we can contribute? And after all, we are only mappers of using the data that are available. So here you see a couple of degradation or land surface indicators rather, like bare area coverage, vegetation coverage and change based on bush encroachment, all kinds of surface features that we are able to map. So the SALDI workflow is based on, of course, the Sentinels, Sentinel-1, the radar satellite, and Sentinel-2, the optical satellite, but not only on the data itself, also on well, higher level products, such as I will show in a minute. But what we also use is, of course, contribution from other data like JEDI, now a perfect set of height measurements from the ISS. We are also using DEMs, elevation models like the Copernicus DEM, or also for Kruger Park, we produced a few years ago an elevation model based on a digital metric camera. So depending on that input data, we are then able to derive products. So one would be time series analyses, such as breakpoints, more known to the optical community, but also possible to do this from Sentinel-1. Then also vegetation structural retrievals, surface moisture, and I was, of course, let's say, very curious to hear about the impact of surface or soil moisture on the vegetational development, and then the same for Sentinel-2. And of course, there is a huge list of indices that have been developed starting with Landsat and then continued with Sentinel-2. And all of these products are part of a data cube, and this is actually my main message, that we have a data cube and we invite you to work with us exploring those results. So, but the really exciting part is starting, well, now since about last year, merging these products, seeing whether we just throw in data into machine learning algorithms, see what we get out, or try to come up with more physical-based, let's say, ideas of merging what we see from a radar product or from vegetation. So this is why we call it also categorized surface moisture, and at the end of my talk I show you why we call it surface and not soil moisture. And this, I hope, leads to many discussions with several of you, perhaps lunch break or evening. Okay, so now why can we do this? I have to show this slide because it's due to the Copernicus program of the European Space Agency. And these are all operational satellites. So we have Sentinel-1 and 2 with a 10-meter spatial resolution, something never preceded before. And operationally, we have these data sets since 2018. So we have stepped into a new era of remote sensing. We have higher temporal resolution because we have a series. We have Sentinel-1A and B and C is being launched soon. We have Sentinel-2, the optical satellite. A and B and C is maybe even already up or will come up. The European Commission and European Space Agency have guaranteed until 2040 to have these satellites in space. So whatever we develop now, it's for the coming decades. So this is also fun. Okay, now I do my 4 slides, 10 seconds, introduction to remote sensing. So, I mean, most of you are familiar. So you know optical is here we have the sun, we have the atmospheric windows, this is where our channels sit. So from there, what do we see? Okay, we measure reflectances, vegetation, many things, water, impact, all of this. But of course now this is where we are in the visible and infrared, near or middle infrared wavelengths. Also hindered by clouds, of course. Now, looking to radar, we don't need the sun anymore. We are using microwave wavelengths. Everybody who has a microwave in his or her kitchen has your own little microwave sensor, by the way. And so knowing how a kitchen microwave works, you could already interpret radar images because radar reflects water. So whether it's water molecules in the vegetation or in the soils. So that is what we can measure, and we are independent of sun. Now, this slide is really important when we watch radar images. It's something totally different. That's my 4 slides. Yeah? The message is it is very different from what you see in optical wavelengths. You are now looking into we call the scattering media. You're looking into the vegetation and how much water is there in the vegetation. This is why we call vegetation a water cloud, actually. There's famous publications. Okay, so the wavelengths we are using are 5 centimeters for the Sentinel-1. There is also operationally, but unfortunately, unfortunately right now commercial, may change next year with American Indian NISAR system, wonderful, L-band 23 centimeters. So these radar sensors reflect how much water is on the surface in the size of the wavelength. Okay, so now I'm coming to the results, some products. One that we were really proud of is a Slangboss monitor. But why I'm telling you this, because this was outside the parks, we saw that when we use this Sentinel-1, I'm not going into details here, backscatter, let's just see backscatter. Yeah, we have a nice time series, as we've seen before from the optical satellites, but we can also use interferometric products that even are more sensitive to the structure of the vegetation. So with these time, here you see January 15, that's when Sentinel-1 started, so we have many years of data, and yeah, the mighty time series. We could develop an algorithm that separates the Slangbosch from the grassland. So in this was for us a very helpful basis to what Nangula is doing now in Mokala National Park, to come up with what we call like a scattering unmixing method to understand what is the scattering from grassland versus bushes versus trees, because these are the three composites of a savanna, and we want to monitor changes like we've done here in the Slangboss. Now we have a map of when we measured Slangboss where. Yeah, so this is, of course, this was towards Slangboss, and the same is being done for Mokala, but for other surface types. Another example I presented last year, so just quickly, is a woody cover change. Again, radar is sensitive to structure, and you can build up a semi-empirical regression method, well, either using actually Sentinel-2 as well, using this fabulous JEDI LiDAR system. Yeah, this is the best in situ training method for us available. So then you can come up with change intensity, because, but this is a total different discussion, we call this woody cover, but is this really woody cover what we are mapping? Marco, our PhD student sitting up here, he's right now trying to develop something between radar and including optical products to come closer to whether it's really woody cover or maybe just a cornfield with a lot of vegetation volume. Now, last point on the slide, soil moisture. And you all recognize here Kruger Park. This is the location of our soil moisture instrument in Lower Sabi, and thanks to Tersha for helping us with all installations and keeping it running. And we will look now at a small animation about this region. So we have Lower Sabi rest camp, and then we go here over the granite basaltic border, and it's a rectangle looking here into this region. Now on the left side you see the NDVI of Sentinel-2 over, and the time frame is 2019 to 21. In the middle, you see a burnt area index, and on the right side, you see the Sentinel-1 backscatter. And if it would be a bit darker, then you could see it better. Okay, but it's not exactly the same dates between Sentinel-1 and 2, but around a few days on and off. Yeah, but I'm showing you just to show you this power of time series of all these surface features that are changing, and you can map this now in a weekly fashion. If there are no clouds for Sentinel-2. With radar, you get your weekly data. And you see the burned area index showing the burned areas. It's reflected in the backscatter as well, but the backscatter is, of course, also related to moisture, surface moisture. Now, coming to this example, surface moisture, the models behind it, and there is a global CCI soil moisture, it's called, a global product, they are based on giving a wet reference and a dry reference, and then develop a semi-empirical model and retrieve something what they call soil moisture, because the models think they are kind of taking care of this slow development of the vegetation features, which is not true. So as we can show here for Kruger National Park. I just first go here. So these are now our soil moisture products, and we changed to surface moisture now. This is for the 30th of December. So for each of the acquisitions of Sentinel-1, you can develop such a map. It's just the result of a model. So 1st of January, 7th of January, etc. So for each of the acquisition dates, you get such a result. So now we can train the model and we can validate the model with this wonderful in situ data. And well, I'm so happy that we did this 4 years ago to deploy those sensors here at Lower Asabi, because the reaction of the soil moisture, and here the instrument was flooded. This is Corona times. Until we came back, we had to repair it. This is, well, things happen. But now it's working nicely again. They help us to understand, is this really moisture what we are seeing, or what is the impact of the vegetation on top of it? Because now looking, and most of you are familiar with this, this is a very wet year with a lot of vegetation, and this was after a severe fire in in September 2020. So what happens to the surface? And now the radar person, again, looks at this image and looks at the surface. What this means, this is a very smooth surface, almost very smooth soil particles kind of clogging the surface, and also then rain coming on top of it. So it's smooth. It's causing a very different radar backscatter than what we would think because of the soil moisture. Now this is really complicated, but you get the graphs anyways. Let's look at the brown line. Here the brown line is our in situ increasing in situ soil moisture because it's starting to rain. These are the precipitation events. Same thing here. Yeah, but now if you look at the blue line, that is the global soil moisture product. Yeah. It is not really increasing when soil moisture increases. Then it goes up a bit when this is the NDVI. When NDVI goes up, that soil moisture index goes up. But now what happens in that year after the fire? The global product is not increasing, and the purple line is our soil moisture, rather surface moisture product. And you see here it is increasing with the in situ, but here it stays low. And why is that? It's because of that scattering mechanism at that surface. So this brings me to machine learning is fine, but you need to understand these physical interactions on the signals. Whether it's optical, it's more tempting with optical than with radar perhaps, but you need to know your surface very well. So, and this is now what makes me as a remote sensing nerd really happy. Yeah, we got all of this together in the data cubes for each of our 6 sites. And just last semester, Marco was the teaching assistant in that class. We had master students, some of them no idea of remote sensing or very little, no idea of programming, and we made them familiar within a few weeks to use the Jupyter notebooks, use the data cube, and they came out with really nice results. So again, what's next? We have many, many friends in South Africa now through all the installations and traveling. But I want to invite you to cooperate with us. If you are interested in one of these regions, better even come to us to Jena and work with us on the data cube. And I think that's my end. Not forgetting who all contributed to this from Sun Parks. You see the section rangers, the game guards, and of course the scientific staff. So thanks to everybody. Thank you very much.
Thanks so much. Yeah, please meet up with Chris at tea time, and tonight there's some really exciting data sets and data cubes to work with. Next up is Sam Harrison. Thank you, Sam.
Hi, I'm Sam. I'm a researcher from the University of Edinburgh, and I'll quickly run through some of the work I did during my PhD on mapping tree assemblage gradients in forest agricultural mosaic landscapes with satellite remote sensing. So agroecosystems we know can support good levels of biodiversity if they're managed to do so. And one of the key ways this is done is by planting or maintaining trees on farms. But at the moment, there's an unmet need to monitor aspects of biodiversity and ecology in these landscapes. And we all know that satellite remote sensing can be a really valuable tool in this, and for monitoring things at landscape level and beyond. But so far, this is rarely applied to agroecosystems. One way that's a novel way that's being explored to monitor aspects of biodiversity and species gradient mapping in particular is this idea of Earth observation ordination mapping. And again, untested in agroecosystems and rarely with open access data. So it's not useful at the moment as a kind of operational monitoring tool because of cost and prohibitive data. So what my research tries to do is apply this method to these untested landscapes with this data. So we collected tree inventory data from plots in 25 kilometre square landscapes in eastern Uganda, western Rwanda, and in central Honduras as well, to get a stringent test in 3 different agricultural landscapes. And we used a combination of optical and radar data, both L-band and C-band radar data, and some auxiliary data as well. That might be useful in explaining some aspects of community composition. So the method is essentially you start with an ordination, in this case an NNDS, and this explains the kind of the gradients of change in community composition within that inventory data. So plots closer together in ordination, more similar, plots further apart, more dissimilar. We then extracted the axes values of all of those points and did 2 models, one for each axis, where our NMDS values were the dependent variable and our remote sensing data was our predictor variables. We then ended up with 2 maps, which we can display as red-green in a single map. So each pixel has a predicted value of where it lies in that original ordination. And the bottom right is kind of a key for an output map. So the pixel colors, according to that key, are where they lie in that ordination space. So quickly running through the findings. First of all, it works even in these really complex landscapes. These are predominantly smallholder farming landscapes. And we get some really promising results. The second is that this fusion approach using optical, different radar data is really important because in our models we see that the band importances really vary between axes and between sites. So the gradients that are being explained by different data in each site. And thirdly is that this method provides a nice nuanced picture rather than our kind of classic classifications which give really discrete boundaries. We get a more nuanced picture of assemblage gradients. And I'll just quickly run through an example from Uganda. So on the left we have the ordination, on the right we have the output map. You can see this gradient from orange through to yellow, and this is the agricultural landscape. And if we look across the ordination, we can kind of pick apart what species might be making up those landscapes. So in the orange, we have species like avocado, jackfruit, mango. As we move up to the yellow pixels, we're getting species like Korea and Combritum in that landscape. We also maintain the discrete boundary with the forest, and you can also see gradients of change within the forest. Again, looking across the ordination, picking apart what species might be making up those colors, we think there might be some sort of aspect of a disturbance gradient being picked up by those color changes. So yeah, just to wrap up, those are the findings, and the method was successfully applied to multiple agricultural contexts. So I think it's a useful tool to think about when we're thinking about monitoring these landscapes. That's me. Thank you.
Thanks, Sam. Very interesting approach. Sure, people will catch up with you over tea. Next, we've got Lucy, and we're back to restoration and using drones. Looking forward. Thanks.
Hello, my name's Lucy Wilson and I am very excited to share with you today a paper that I published last year on mapping the restoration activities on Dirk Hartog Island using drones. Dirk Hartog Island is located in Western Australia. It is a coastal rangeland, which means it consists of grasses, forbs, shrubs suitable for browsers and grazers, and some intermittent trees, which unfortunately all my photos I took in the field of trees were just of singular trees, but you can imagine that there would be trees dispersed throughout this image usually. The vegetation structure in here is very similar to the African savannas that we're discussing this week, and the project had secured 10 years of funding, and they knew they wanted to use drones for their long-term conservation monitoring, but there was some uncertainty about how to best approach this and whether drones would even be viable for this purpose. So we set out to solve this by asking 3 questions. Firstly, which variables would be most suitable for a species level classification? What would be the optimal flight height for the drone to perform, given a trade-off between resolution, accuracy and flight height? And also lastly, how can remote sensing analyse and detect plants using machine learning, and what accuracy can we potentially obtain? To do this, we set out in the field and did drone monitoring. We surveyed 2 sites, one with high species diversity consisting of 9 species and one with lower species diversity of 4 species. We captured very high resolution multispectral imagery, and yes, that is 5mm pixels. We also ground truth plants with an aim to capture at least 10 samples per plant species. And we did an object-based image analysis, which you would have just heard from Julia. Now, usually with plant identification in object-based image analysis, you'll bound that object to the plant canopy. But we can see in rangelands where shrubs and species usually coexist in space and intertwine that this isn't always feasible. So instead, we modelled our object to be closer to the leaf size rather than the canopy boundary. And then lastly, we did a machine learning algorithm to classify plants to a species level and then tested the accuracy. So what we found from our first question about which species would be suitable, you can see here, do I have a laser pointer? Yes. On this left column, we have study site 1. On this right column, we have study site 2, which shows the variable importance in the projection scores. And we found that variables that fell into the reflectance, greenleaf algorithm, spectral reflectance intensity and height was suitable. However, object-based image analysis variables such as texture and shape are usually argued as a strength, but we found they weren't as valuable for identifying plants to a species level. This is our accuracy outcomes. We use the Cohen's-Kappa coefficient to measure accuracy, with red squares being high accuracy and blue squares being low accuracy. We have our pixel size on the x-axis, ranging from our initial capture at 5mm up to a 50mm pixel resampled, which reflects a higher flight altitude. And then we also have our segmentation scale. So in e-cognition, we could control this, which controls the shape of the segments. And we found that a 5mm pixel accuracy could be just as accurate as a 45mm pixel. So a lower flight height might not necessarily result in high accuracy, and a higher flight height can expedite your surveys and cover a much greater area. And you can see this in this trend region here. And lastly, our accuracy outcomes. So we did find we could achieve a higher accuracy in our lower species diversity site. 70% and 53% overall accuracies for left for study site 1 with a higher species diversity and right for study site 2 with a lower species diversity. And now we knew we were ambitious when we set out in the field to try and do this level of classification. We do believe that this can be improved on with some post-filtering or grouping species into similar growth forms. And if anyone would like to come discuss how this might be applied to African savannas, I'd love to speak with you, and there is a link to the paper here as well. All right, thanks.
Thank you very much, Lucy. Next up is Lorena, I think from Edinburgh as well. Thanks, we're looking forward.
Hello. So this project is a collaboration between the Zimbabwe Forestry Commission and CIOSA. And if you don't know what CIOSA is or you want to learn more about it, I also have a poster I'm presenting this afternoon on it, so come find me there. And so this project began out of a workshop we held a year ago at the Zimbabwe Forestry Commission, trying to analyze all of the wonderful plot data they've been collecting. And what we quickly found was that the vegetation class map that they had been using did not match their plot data. We were getting plots that were classified as Mopani that had absolutely no Mopani in them. So we knew immediately there was an issue. And so we have attempted to create a new vegetation map for the country that hopefully reflects a little bit more of what we're seeing in the plot data. And so what we did is we classified the plots based on indicator genera. So we assigned them to one of 6 classes based on these genera, so it had to be at least 10% basal area to be attributed to these. And so initially what we did is we analyzed the plot data just to look at what these vegetation types are doing in terms of structural diversity, species diversity. What we found is that there are more structural differences than there are floristic differences. So here I have figures for the structural differences. You can see Baikea and Miombo are coming out as having much higher AGB, stalking density, and height, but there were no differences in the species diversity metrics, though there are some differences in patterns of dominance. We also did a species ordination to look at if the classes that were in the original map are really distinct floristic groups. We found is that there's a lot of overlap between these classes based on our plot data, although Mopani and Miombo appear to be distinct, and Baikea has a huge amount of overlap with the Miombo. And so using that data, we incorporated additional ground truth points and satellite data and trained a random forest classifier to create this map of the country. And so we're getting a majority of Miombo with Mopani coming in second in terms of cover, with very little Baikea or Acacia. But of course, most of Zimbabwe isn't actually covered in these vegetation types. And so what the map probably looks like is a lot more like this. So removing the low biomass areas that are likely converted lands, this is probably what is an estimation of what the cover looks like, with some additional metrics for carbon based on McNichol et al. 2018. And so we're getting about 14.5 million hectares of wooded lands. So the next thing that we're interested in looking at is how these vegetation classes are changing over time and which ones are changing most rapidly. So using the biomass change from McNichol et al., we're looking at which areas and which classes are gaining and losing biomass. And so we are seeing quite a bit of gains and some losses, but there's actually, in this preliminary analysis, a net increase in biomass across the country. And if you convert that to area, it's a very marginal increase in area But once you break that down into the different vegetation types, you actually see that you're losing some area in Baikia and Miombo. And because Miombo is the largest class, a less than 1% loss is actually quite a bit of area. And just to end, so this is just a very preliminary look at what's happening in Zimbabwe. But what we're finding is that our estimates are a huge change from the estimates that were made in the 1990s, and our estimates are also a little bit lower to FAO and Hansen estimates. So biomass might be marginally increasing, but there are certainly some classes that are under threat. Yes.
Thank you very much, Lorena. Okay, we don't really have time for questions, but meet you at a poster as well this afternoon. Next, we've got Ashwini Pandey and talking about using remote sensing approaches for mapping invasives in Kruger National Park.
Hey, good morning. I am Ashwini. I'm doing a master's in machine learning and artificial intelligence at the Stellenbosch University. And today I'll be talking a little bit about my project. My supervisor is Sandra, and co-supervisor is Pietro. And cool. So essentially, in this particular project, what we're trying to do is see if we can use aerial photography to identify invasive species. And obviously, they've got a major impact on the biodiversity within the Kruger National Park, I think, as has been covered extensively in various talks. So I won't cover much of that. But essentially, the idea in this particular one is, how do we sort of use computer vision to essentially hasten the process through which we can then start to act and create the appropriate responses to deal with it. In the two species that I'll talk about in a second, I think the main thing is more about containment, but potentially, how could we, for newer invasive species, how could we identify them early on enough and approach a different thing for that. Just to sort of put this work in context, so this is a smaller piece of work in a larger study that's being sort of initiated by the Czech Academy of Sciences and by Peter Peszek. I hope I haven't messed that name up. But yeah, and Llewellyn Foxcroft as well. And essentially, it's a multi-year, I think about 3-year project where there are 3 invasive alien species that are being looked at. And the idea is to try and see if we can map them and to what extent they have disseminated across the park. And if essentially then try and look at it temporally as well once we've got a baseline for their spread. It's being largely studied alongside on several sites, but along the rivers. My particular focus will be around the Letaba River. Cool. So the two sort of species that I'm focusing on is the Datura and the Xanthium. I've put in these images just to sort of emphasize the point. So if you look at the top 2 images, they obviously are quite tricky to distinguish, and I think that's part of the point. And the bottom 2 images are obviously the clearly distinguishable ones, which were taken by Sandra, I think. So yeah, I think when it comes to computer vision, I'm more of a data scientist. I'm not really an ecologist. So to me, the the differentiating factor is kind of key from an image perspective, because that's what I'm going to rely on to be able to look at that. Cool. So in terms of the data, we are focusing on aerial photography. I think that's in some sense it's good and bad. I think with the satellite data, it's nice to be able to segment at a macro level, but I think it starts to become a bit difficult to try and identify species with that level of data. So in that, the aerial photography is excellent. I think it's perhaps not maybe as detailed or to the same resolution as maybe a drone can get potentially. But we do have quite high resolution data up until the 5 centimeters. And essentially what you're looking at is that same plant in the different resolutions, and it very quickly starts to get quite messy. And so what we're trying to do in this project is go from the image on your far right and try and see if we can get to the image on the far left, firstly by just essentially looking at foliage, etc., segmentation, and get to that component. And then once we've gotten there, try and classify if this is a xanthium or a datura. And that's primarily going to be based on the leaf structure. Cool. So in terms of the preliminary analysis, sorry, one sec. Cool. So what part of the work is also to try and make it reproducible, which I think a lot of folks try and do. And in that effort, the data is quite large, and so that needs to be stored somewhere that can contain it. Computer vision, by its very nature, uses a lot of compute resources, which is another thing that needs to be catered for. So essentially, I've chosen to do this on Google Cloud, which helps us with storage and compute, and do a lot of the work in Python, and do it in a pipelined fashion, so that when we get newer data, we can easily sort of go through it and at least get to the baseline that we intend to. This is just a sample plot of the Letaba River, where essentially what we've got are some labels. So as you can see, this is specifically for xanthium. So the gray dots are those labels, and then the green dots are sort of other plants. And these are, this is at a low resolution, and just to sort of emphasize how it can get quite tricky to work with this, this is when you're cropping it, that's what we see. The idea is to run this on the higher resolution and get closer to what you saw earlier on the left-hand side. One of the tricky challenges with these crops is that when you sort of look at the points that have been labeled manually for what the plant is, it's sort of a point buffer, or it's a point, and we need to create a buffer around it, which is a little bit of a tricky component. So in terms of like not including too much, but getting that mix right. In terms of the approach that's going to be taken for this project, it's essentially getting this raw data that we have from the field into smaller sort of labeled chunks, which is what I've just shown you. And at this point, essentially do segmentation, not of, it's not necessarily of the highest accuracy per se, it's just to get us into smaller patches of the image so that we can focus on them. And then from that, we want to be able to get to images like you saw of the leaf. Sorry, let me just go back quickly. So if you look at this, and from this essentially generate a bunch more labels for this particular plant by segmenting the leaves. So I think that's this component here, because at this point the labels that we've got are obviously for the location of the plant, but not necessarily at a leaf level, and then use that data then of the leaves to classify the species. There's the last aspect of this, which I think is going to be particularly challenging, which is with any sort of computer vision project, which is once you've got a ton of images classified, someone has to manually verify that, which will require some degree of domain expertise and a lot of time. The challenges that we sort of have in this, in terms of collecting the data, I think there's two components to that. The aerial photography, which is, yeah, I mean, it's got its own flow of complications, but yeah, we've got that data, but also the on-the-ground labels for the plants, that's limited in this context, which we then need to expand by generating labels. And the storage and transferring of data, which I think Sandra will allude to in her talk, is quite a challenge. It's quite a lot of data, and transferring it hasn't been easy to be able to get it onto the cloud. The next challenge that we've had is pre-processing it prior to the machine learning. So not the code component, but prior to the code component, there's a lot of different pieces that need to be put together in terms of geospatial component and the images. And yeah, so we've got the limited labels that I've spoken about, and with the processing, I think we've mitigated that by sort of moving the project onto cloud and yeah, the verification of the results that I've mentioned. Cool. Yeah, I think that's it. Just a major shout out to the biomath group from Stellenbosch University. I think there's a bunch of folks speaking from there. So yeah, thanks for the opportunity. Thanks.
Thank you very much. Yeah, as you would have seen, we're running a bit late, so if you don't mind, I think we're going to do questions afterwards and people can catch up with you. So we're going to be going into another sort of subsection of this session, which is focusing a bit less on remote sensing, but on modeling method advances and big data and data sharing. And Sandra is going to start it for us. Thank you very much, Sandra.
Can everyone hear me? It's good? All right. Thanks, Isak. I'm Sandra McFadgen. I'm with the Mathematical Biosciences Group in Stellenbosch, as Winnie alluded to. The bio actually stands for biodiversity, as we're particularly interested in using an interdisciplinary approach to tackling some biodiversity-related questions. So as Winnie said, there's a lot of us that are not ecologists. I'm probably one of the only ecologists that is actually in the setup, but it's a really exciting time to work together and to look how things that have been around for ages in maths, for example, or in physics, can actually be applied to complex ecological problems. And in this new information age, I think biodiversity conservation and research in general is rapidly changing. And the new currency is data. And we're all drowning in it. I mean, as technologies advance, information is more easily accessible, And it's also easy to publish that data, but it often goes unchecked. So the data that you download, you're not always 100% sure where it comes from, what is the quality, can you incorporate it into your own data sets and use it in a comparable fashion. So we have exponentially more data or information, but we don't have more time to actually filter and assimilate it. I'm sure you're all familiar with the idea of being able to catch up, keep up with the literature. It's becoming more and more impossible to do so. And just when you think you're on top of something, something else comes out in the literature and you think, oh, that was rubbish, I need to now move on to something completely new. So it's quite a challenging time to be a scientist as well. And in South Africa specifically, we often don't have the resources or the skills available to actually work with these large data sets. And there's a lot of technology out there, Google Earth Engine, to name a few, but we don't always have those skills, especially as ecologists. And although one might think that more data should be a good thing, more information leads to more awareness, which leads to more action. Especially in this current biodiversity crisis that we're sitting in, where a global concern can actually only be mitigated through local actions. But the data needs to be accurate and reliable. The old notion of garbage in and garbage out. How do we filter it? How do we filter out the misinformation? And how can we standardize quality checks, data formats, improve integration and encourage data sharing? For local initiatives, this is especially important when it comes to being interoperable. Like you want to be able to combine your local data set with a global one. You need to have it in a format that is transferable so that you can look at longer term trends at a local scale. But we need, as I said, we need the necessary skills and resources to successfully generate, collect, store, share, analyze, and promote synthesized biodiversity data. And that is a skill that's not generally associated with conservationists or ecologists. And herein lies the paradox of modern ecological research. Oh, my mouse is stuck. In that everyone wants clean, long-term, quality-checked data sets, but few are actually want to or even able to actually collect, clean, or manage it in a standardized way. And remote sensing has become really popular because of that, because you can just literally go onto Earth Engine and download 50 years of data and you can start analyzing and it's all comparable. But this also puts some of the longer-term field monitoring programs at risk in many ways. And I've heard of a lot of protected areas who are struggling now to keep these long-term monitoring programs going because they can't rationalize using the funds for those programs when you can just sit in your office and download a remote sensing product. But it's important that we as ecologists keep stressing the idea that we cannot go without the field monitoring data. The remote sensing data, we cannot verify that without the actual localized field monitoring data. And not to mention the backlog of data that's actually there. There are gold mines of data sitting in archives, in people's offices, as hard copy data sheets. And those are invaluable data sets, but when you come to Africa, no one wants to sit and digitize a data set. They want to follow the elephants or measure the trees. So we need to try and generate awareness in this and try and make it appealing, save a rhino, digitize a data set, something like that. I'm not sure how to do it, but we need to start thinking about that. And this is where we enter the field of biodiversity informatics. In this information age, it seems obvious that we need to dive into this world of informatics. But while everyone in this room is probably well aware of what biodiversity is, informatics might be a little bit less well known, in that it's the study of the computational systems, especially those for data storage and retrieval, and it is synonymous with computer science, where the notion is that basically the transformation of data into information. So in combining this, we can look at biodiversity informatics, which applies these techniques from information technologies to improve the management, presentation, discovery, exploration, integration, and analysis of biodiversity data. And it's a multidisciplinary problem, and it can lead to the unlikely pairing of disciplines. It's almost the punchline to a bad joke. But we really need to start putting more effort into establishing these transdisciplinary collaborative networks that bring together expertise of computer scientists, information technologists, librarians, historians, mathematicians, putting them all in the same room and creating a dialogue, but also starting to form a new language where we can actually understand each other. And the idea is that ecologists should actually stop toiling in this unfamiliar data science domain. You don't need to do it alone. You just need to work together so that we can develop this new understanding and have the time to sit and listen, because I think that is the tricky part, to actually listen to a computer scientist or a mathematician, and for him to listen to you. I mean, ask Ashwini. I think he hated me in the beginning, but I'm starting to convince him otherwise. And we need to develop this mutual understanding and respect and trust, which takes time. So in an attempt to crystallize some of these ideas, last year we brought together a group of key biodiversity stakeholders with distinctive expertise to help develop a way forward for biodiversity informatics in South Africa. And we wrote this paper in Biological Conservation, which comprised 37 co-authors from 20 different departments across 10 different South African universities, 7 national and provincial conservation agencies, and various different institutes from private research, from private conservation research and management bodies. And the article reinforces the importance of linking these local systems with worldwide networks to develop a more integrative resource for holistic knowledge generation, especially when it comes to biodiversity declines that we're facing today. And at the same time, we went and put a list together of all the challenges, but also the opportunities and innovative solutions that biodiversity informatics is facing in South Africa. To start with, we broke the whole system down into a sort of pipeline, into the core elements that we thought made this up. And it started from a data in, where the data flows in, have a data management protocol linked to a data centralization. The information is then processed into a digestible format and then is provided as a service to researchers and decision makers. And then outside that system, we touched on a few of the research development goals, the knowledge that is synthesized from that, and the implementation that happens, with the goal of an understanding coming out of that system in the end. And one of the things, for me at least, after we wrote the paper, I realized 37 authors is not nearly enough to actually capture all the needs from this point of view. So we then hosted a biodiversity informatics workshop at the end of last year. We had about 100 delegates, which still isn't enough. But we had discussions that were centered around the 6 keynote addresses, 34 topical presentations, and probably most useful was 3 facilitated panel discussions, where we actually paired completely different people together in the group so that we could hear the voices of a data scientist, a mathematician, an ecologist, a manager, a policymaker. And those yielded some really interesting results. And overall, we saw there were many positive developments that have actually occurred in South Africa already with biodiversity informatics. The consensus was that we still needed more investment into better support for environment protection and monitoring, and to improve communication with key stakeholders and policymakers. And we went into the process thinking of a biodiversity informatics hub, and it morphed more into a biodiversity informatics network. And some of the key needs that came out of that was to divided into people, portals, protocols, practice, projects, and patronage. We just had to have P's, I'm not sure, but it sounds great. But essentially, we need to nurture a more inclusive community of practice around biodiversity informatics, and we shouldn't reinvent the wheel. We've already got systems out there, so the way that the network needs to be built is to rather draw on information sources that are already there by producing reproducible workflows and APIs that you can pull from existing data sources and then feed it into workflows that are tailor-made to different scenarios. So I'm not going to go too much into that. What I wanted to talk today about was, as a precursor to these aspirations, we started working on a how-to guide or a toolkit for ecologists and conservationists or protected area managers to design, develop, and maintain and serve in-house biodiversity data. So one of the big things that came out of the workshop itself was that people generally in South Africa are mistrustful of putting their data sets in large global repositories, and trust needs to be built before things like that can happen. So it seemed the best approach would be to create a system that anybody could use offline, but with the view of it being able to go online at some point. So rather than forcing you to upload your data onto an online system, we create the infrastructure or the design of that architecture to be downloadable. You can use it in your own institute, but then the standards of your data are set up already to be able to transfer back to a larger online system if necessary, but only once you're comfortable to do so. You can't force anybody to give you their data. And this is what the basic framework looked like. We divided it into these 4 stages. And as I said, we put a big emphasis on interfacing with existing systems, so pulling data from established online sources. And it has a repository for all these reproducible workflows, so things like GBIF or APIRS or NASA's data sets. You can go on and take the workflows and just change your study area of interest, and it will extract the data for you. And otherwise we try to unpack what are the different data sources that we were interested in, and drilling down into those, you can see that they're wide and varied. And it can be difficult to navigate such a heterogeneous data landscape, not just in terms of the data formats, but also the people that are housing that data and maintaining that data. So essentially, we try to keep it simple, and we broke it down into the bare minimum file structure that we would need to house these spatial data sets. But at the same time, we sit with the same situation where I mentioned in the beginning, all these long-term data sets that have remained uncaptured. And we wanted to have a way that we could automate their digitization, where we could automatically scan them in and convert them into a digital format. And for me, at least, I think that alone would be a really big contribution to conservation agencies in South Africa. So I played around with some machine learning, thanks to Mike. I think Mike is somewhere here, maybe. Mike's got this amazing long-term data set. A lot of it is sitting in hard copy format. And we took this data set and a few of the options that programs that we had from that, and I looked at using some machine learning, specifically open source computer vision and machine learning software libraries, to see if I could extract the data from hard copy sheets and tables. Obviously, the typed versions. How can it go from 1 minute to time up? So obviously, the typed version it works really, really well. But then ecology is not that simple. So when you have things like this, it's a little bit more tricky to automate it, but we're getting there. And at the end of the day, we will have something that you can use that will automate it as far as possible. But is this achievable? And that's the slide I want to leave you with, is can we actually do all this that we're talking about? And we've got some exciting research programs that are happening this year. We've been funded by the National Institute for Theoretical and Computational Sciences. Our project is Advancing Biodiversity Informatics and Ecological Modeling, linked with another program. And we're going to be holding a hackathon to try and address some of the ecological questions using different types of thinking in terms of computer science and coding and mathematics. And then we've also got this big EU Horizon project that just kicked off now the 1st of March, the biodiversity building blocks for policy. And it's got a team of 13 different institutes across 10 different countries, of which South Africa is one. And the B Cubed is going to develop the infrastructure and workflows, tools, knowledge products, and indicators for biodiversity monitoring at scales that are tailored to the needs of decision makers. And Steve says, no, that's not going to happen. Okay, so it just kicked off on the 1st of March, so I'm really excited about that. And we have a PhD post that's linked to this, so if anyone has an aspiring PhD student that you'd like to come and chat to me about, please do so. And yeah, I think this is an exciting time to be an ecologist, and yeah, watch this space. Thank you.
Thank you very much, Sandra, and thanks for being one of those ecologists walking into the bar and making some friends in there. So we've got two 5-minute presentations remaining. So Taseyiwa Mumbe will be talking to us now on using terrestrial LiDAR to map biomass of trees in savannas. Thanks so much.
Okay. So good morning, everybody. My name is Taseyiwa, and I'm a PhD student at the University of Vienna. And together with my co-authors, we looked at estimating the above-ground biomass of savanna trees using terrestrial LiDAR, because we wanted to employ a non-destructive method. So to do this, we used terrestrial LiDAR together with volume reconstruction algorithms that I'll talk about next. So what did we do to achieve this? So we scanned a 6.25 hectare plot at Skukuza Flux Tower in September 2015. And using these quantitative structure models, we were able to estimate the volume of the trees. So what are these quantitative structure models? So they represent trees as collections of cylinders. And then from there, we can extract various tree attributes. And these models have been used with success in different biomes, but they haven't been tested in the savanna biome. So that was our main objective. So from there, the above ground biomass that we got using these reconstruction algorithms, we compared it with allometric equations that are currently being used in the savanna. So the methods that we employed, so from that scanned 3D image that I showed you, the top video there is now showing you all the segmented trees. So we had to do a process of segmentation where each and every individual tree in the plot was given a unique tree ID. And then the image on the bottom is just showing two segmented individual trees. So these segmented trees are the ones that we then now reconstructed to get the various tree attributes that we were interested in. So I'll just show you an example. So Fig 3A there is showing a tree point cloud, which has been segmented. And then the figure 3B is showing now the resulting QSM model with the various tree attributes that we were interested in, like total volume, branch volume, diameter, and breast height, tree height, and crown area. So what were the main findings that we found from this study? So we found that using these volume reconstruction algorithms, we can accurately estimate the biomass of savanna trees. And then looking at figure 4, which is the regression model comparing our AGB from the QSM model versus the allometry, we saw that using allometric equations, we have an underestimation of the biomass, especially in the large trees, mainly because some of these allometric equations are only developed using just DBH or tree height. So we do not account for all the biomass that is stored in the tree crown. And then also one of our equations used crown area as a predictor variable in estimating biomass. And we saw that this is the allometric equation that gave comparable results as compared to the QSM model. And this is in line with other research that has been done, that when you include crown area in the development of allometric equations, it estimates the biomass better. So from these results, what can we conclude? Because we managed to reconstruct a total of 969 trees from our 6.25 hectare plot. So with this kind of data, we are now looking at developing accurate tree biomass equations for the savanna ecosystem using high resolution TLS data. And as you can see from my animation, this quantitative structure model can also reconstruct even multi-stemmed trees. And then finally, we can also quantify the gains and losses in the savanna ecosystem by the application of multi-temporal TLS data sets. Since savannas are very dynamic, we can now quantify the changes caused by fire, by herbivory, by even drought. So lastly, I would like to thank all the funding partners, my supervisors at University of Vienna, and all the co-authors. Thank you very much.
Thank you very much, Tashiwa. That was a great presentation, and you stick perfectly to time, so well done. Yeah, just to remind you, there's quite a lot of posters associated with the session, so just check your programs. And the final presentation for the session is by Peter Scoggins, talking about rethinking woody plant functional types in African savannas. And I think that specifically links to your and Greg's poster as well in the savanna models, if I understood it correctly. So thanks, Peter.
Yeah, I also kind of feel a bit like the ecologist walking into the bar full of modelers. Normally at these meetings I would be in something like the plant herbivore session or something reporting on results of some other experiment that I've been doing. So it feels a bit strange. Greg's tried to convince me a number of times that I've transitioned into being a modeler. So I've spent the last 8 months or so harvesting data from all the experimental papers that I could find to come up with this. Yeah, so it's like the reason that I transitioned into this kind of space was because of a question that I've had for quite a long time, is that can't be addressed through regular kind of field experiments or lab experiments or anything like that. It's like, what happens if you remove things like condensed tannins out of trees? What's going to happen? You can't do that easily in a physical experiment, but you can theoretically do it in a virtual experiment in a model. Greg, some years ago, had the task of parameterizing the Savanna ecosystem model for this park. He used 5 woody plant functional types. For those of you who don't know me, my main interest is in trees and browsers, so that's why I'm focusing on woody PFTs and not others. Those are the 5 there. It's a pretty subjective classification, but that is the kind of traditional knowledge that has existed in savanna science for many decades, so it was kind of the logical thing for Greg to do at the time. We've heard quite a few talks already in the last couple of days about the scarcity of data, the lack of basic ecophysiological data for plants, stomatal conductance data, things like that, which there's tons of it for temperate species, tropical forest species, but very little that you can find on savanna tree species. So basically, when Greg shared some of the Savanna code with me about 1.5 years ago, and I started looking at the PFTs and their parameters, I thought, well, this is all rather subjective. Let's try and make it more quantitative. The problem with subjectivity, like if you ask a bunch of people like us, I'm going to take more than 2 minutes to finish this, I'm afraid. If you ask everyone in the room here if a certain species is a tree or a shrub, they're going to give you different answers. This is a table from a survey that exact kind of survey that was done at this meeting in 2011, asking people what's a tree and a shrub, and some species just get very mixed responses, and some are very definitely a tree or a shrub. So this is the kind of thing that I'm trying to get around. So the first thing I had to do is decide what species and I'm going to start using to get quantitative data on. Looked through a whole lot of literature to find anything that I could find on diet selection of various browsers. I came up with a list of 59 woody species in the end that I could find data for at least 2 browser species. The next question is what is a tree? So I used that article that I referred to just now, which is if the species height is greater than 4 meters, then the species is a tree, otherwise it's a shrub. And I got a whole lot of data from various sources to decide whether which species in my list of 59 were trees and which were shrubs. This also is a bit of an introduction. You need to know some of this stuff if you want to understand our posters out there. The next question is it a broad-leaved or a fine-leaved species? Now, generally, most of the species, most people there would be consensus about whether it's broad-leaved or fine-leaved, but there are a few that I've often heard, like Acacia nigrescens as an example. Some people would say it's broad-leaved, some people would say it's fine-leaved. So I came up with the second rule was that if the leaf or leaflet average size is less than 225 millimeters squared, then it's fine-leaved, otherwise it's broad-leaved. I then, because of my interest in browsers, and obviously my interest is in plant defenses against browsers, and everyone says structural defenses are the big thing, forget about chemical defenses, they don't work against browsers. So I got Tristan's investment in structural defense, unpublished data from him, thanks to him, and came up with this rule: if the square root of the ISD is less than 13.7, then the structural defense is low. If it's greater than that, then the structural defense is high. And I came up with a rule for chemical defense investment of if the nitrogen to ADF ratio is less than 0.1, or if the condensed tannin content is greater than 5%, then the chemical defense is high. And in that way, I could slot my species into a number of new plant functional types that I came up with. And these plant functional types are now quantitatively defined instead of defined in terms of vague concepts like palatability. That table there is on the poster if you want to look at it more carefully and want to ask more questions about it. I then went on to finding values in the literature for all 73 parameters that Savanna needs for each PFT. that's available freely for anyone to download from Zenodo if they're interested in looking at it further. Just by the way, when I did this little analysis last week and drew up this little table here, because the next stage was to put preference rankings onto each of these new PFTs. So I had to find out a bit more about what elephant, giraffe, kudu, and impala prefer. And yeah, interestingly, elephant and giraffe, they quite like the highly chemically defended acacias. The thorns don't really matter to them. Elephant's got a big mouth. Giraffe's got wire stripper-like pliers in its mouth that it can just strip everything off. And kudu and impala basically prefer the less defended species, just as a matter of interest for anyone who's interested in that. And there's a whole lot of stuff there about what we're going to do next. Thanks. And if
Thanks so much, Peter. Thanks everyone for attending the session. I think it was a very stimulating session. It was a long session. We're going to have break for tea now. The good news is we're going to have 15 minutes longer for tea. Well, we're going to start at 15 past 11, so 11:15. But we're also going to take a group photo now, before you fill your cup and empty your bladder. So let's just first take a photo and then you can fill up. Thank you.
presentation is here, they need to load their talk, otherwise we'll just skip that.
Okay, everyone, welcome to this session. I'm Guna Kensani, standing in for my colleagues Sharon, who was not able to make it today. We're gonna get started now, starting with Alison with her talk titled "Genomic Approaches for Environmental Management: The Current State of Play." Thank you.
Thanks for coming. So my name's Alison, and I'm currently a PhD candidate at Charlestown University, which is located in the northern part of Australia, up in Darwin. And today I'll just be giving you an overview of how genetic approaches have been used for environmental management, and also giving a little bit of a plug for my PhD project at the end. So there are a lot of challenges with monitoring. So say you want to monitor something in the environment, like a flora or fauna for biodiversity assessment, or to look for cryptic or endangered species. To do that, you'd go out in the field, collect your sample and identify it. And the current gold standard is morphological identification. So for that, you'd need specialist taxonomic expertise yourself, or to find experts that can do that for you. So you're looking at invertebrates like arthropods. And if you've seen any of my colleagues like Alan Anderson or Francois talk, there are potentially thousands of ants, not to mention other arthropod groups, each with experts scattered across the globe. So this can create a bit of a bottleneck, right? Because people want to monitor this fauna, but there may not be enough expertise to do it, and the supply doesn't meet up with the demand. But what if you don't have to have experts to identify your samples? What if there's a magic tool that you could use to collect a magic tool that you could use and potentially not even have to collect a specimen? Well, it's not magic, and this actually exists today, and it's new genetic techniques that you could use to perform these rapid biodiversity assessments to monitor your target taxa. So there have been recent sequencing advances over the past couple of decades, with sequencing getting a lot cheaper and more accessible. So the human genome used to cost $3 billion back in 1990 to 2003, and today there are companies saying that it can cost $100 to do the same thing. Now, over the past 5 years, there's been next-generation sequencing that has come to a lot of laboratories around the world, and this uses a different technique to the previous older technology, and it basically breaks up the genome into small little reads that overlap, and then you get your genome. To do this, and now you can do this in the field using MinIONs. So they're these tiny little USB-like contraptions that you put your sample directly into, and this could be tissue from any of the taxa that you're monitoring, or a water sample to see what you can get straight from the field. So I'll just go through a few definitions for those that aren't familiar with any genetics terminology. So a barcode is where you have a specific species that you're looking at, so an individual sample, and you take, say, an ant leg, and you submit it to a sequencing agency, and then they will give you a barcode. So they're looking at a specific gene that you'll be able to compare against your different species to be able to tell which one's which. So for DNA metabarcoding, you're looking at multiple species in a soupy sort of sample. And what you get when you do the DNA extraction is you get DNA from your target taxa, so the taxa you're looking at or you want to monitor, and you're also getting DNA from other taxa like your bacteria and your fungi. But when you do the analysis, you get a sort of like when you go into a grocery store and you scan your barcodes, basically you'll be able to identify what species are in your sample. So just to give you a bit of a background about what a metabarcode looks like, finer scale, you have your primers on either end of the barcode and your metabarcode in the middle. And this metabarcode needs to be highly variable between your species, whereas your primers need to be conserved.
Yeah, we can just pause. It's not going to take a long time because we're moving from the generator to the main ESCOM. I just want to make an announcement. There's a talk missing. So if Fraud Forsoy is here, should come and load their talk.
Hey. Cool, ready to go? Okay, continuing on. So just to recap, yeah, you want your primers to be highly conserved and your metabarcode to be very variable. And environmental DNA, or eDNA, you may have all heard of. It's been in the news a lot in the last few years. So all living organisms leave traces of DNA in their environment. So this can be free or extracellular DNA that's shed off of the animals, or cellular or nuclear DNA, which is still contained within the skin cells, but also organelle or mitochondrial DNA from eukaryotes or animals, or chloroplasts from plants. And the organelle DNA tends to be an order of magnitude more abundant in the environment compared to the nuclear DNA, which is still contained in the cells, and that's just due to the nature of cellular respiration. You need a lot of energy, right? So you can get eDNA from different substrates like water, sediment, plants, soil, even scat. So you've seen a lot of poos when you're driving around. You can totally sample those. Invertebrates from pitfall traps, gut contents from inside the animals that you want to monitor, say, their diet, and also, interestingly, air. So there's been a study recently that's looked at using a vacuum to mop up air in a zoo, and they found giraffe, rhinos, elephants' DNA just floating around everywhere. So here's an example of a metabarcoding workflow. So you start off with your specimens or your bulk samples, and on, say, you're looking at insects, you've got a lot of them, and you also have soil or eDNA samples, or also water samples, and they get extracted during the DNA extraction process, and generally the DNA is in quite a low concentration. So what you want to do here is amplify it. So you want to take one DNA and turn it into 1,000, and that's where this polymerase chain reaction amplification happens. And then from there, you take it to a sequencing lab, or you do it yourself on your benchtop MySeq sequencer. And then from there, you compare those sequences to the reference libraries, like BOLD or GenBank, or your own custom reference library, to get out a species list. So here are a few examples of some applications that you can use environmental DNA for. So you can use it to detect environmental change, biomonitoring and biosecurity, which you can use water samples from ports to look at invasive species that are coming in. You can also look at the impacts of industry using eDNA, and difficult to access environments like stygofauna, which are underground in the aquifers that you can't see but are there. Also detecting rare or endangered species, as I mentioned before. Interestingly, for agriculture, you can actually use, you can capture bees to look at plant biodiversity, because the pollen gets stuck onto the bees as well. So altogether, this just gives you a way to rapidly measure biodiversity in the environment. So interest in eDNA has exploded over the last decade. So here's a graph just showing the number of publications across the years. So starting in 2008, it starts off with one publication, and then from 2020, which is the last figure on this graph, it's shot up. And I can tell you in 2022, it has continued to increase. So there are several ways that biomonitoring can be used on a large scale. So there's a movement in Europe which looks at biomonitoring of river systems using aquatic invertebrates to look aquatic invertebrates as bioindicators of river health. And this is called DNA Aquanet. So they've done a lot of work over the past decade developing new genetic tools for these bioassessments. And they're monitoring aquatic invertebrates, but actually these primers are really well, convert really well to terrestrial invertebrates, which is a good thing to remember later. And in Australia, there's the Minderoo Foundation, which looks at oceanomics. So they go around Australia sampling a lot of the Australian coastline, and they have the lab on board, meaning that they can take their water sample, process it on board, do all the bioinformatics, and sort of get a result 4 hours later, which is quite impressive. On more of a local scale, you can use it for monitoring amphibians and fish in water systems that are in nearby creeks and rivers. And it's been found that the eDNA from these taxa have been capture species diversity in much greater detail than traditional techniques. So MyFish is an example of this metabarcoding workflow. And there are also new terrestrial metabarcoding studies being published over the past few years. So some of the challenges with this workflow is the sample DNA that you're getting at the start. So I'll just talk a little bit about that. So some of the challenges are the lack of foundational studies on eDNA characteristics and dynamics. How is it produced? So is it physiological? Is it getting excreted from the organism? What factors affect eDNA production for your target taxa? Also, what state is it found in? Is it small? Is it large? Is it present inside the cell membranes? Is it hard to get out? Will you miss it when you filter it through your water sample? And these two factors can affect the transporter degradation. So the transport is how is eDNA dispersed in your environment? Is it retained by certain substrates like clay, or does it diffuse when it rains? And also degradation. So how is eDNA degraded biologically, chemically, enzymes from microbes breaking it down, and how long is it detectable for? So for example, in some ice cores, you can get eDNA lasting from 1,000 years ago. But if you're looking at soil in tropical savannas, that may not be the case. It may only be a few days. So primer selection is another challenge in this workflow. So selecting the appropriate barcode marker. So for different taxa, you have different gene regions that you want to look at. And so say you've all heard of the CO1 maybe, which was also in Alan's talk, looking at invertebrates. So that is really, really good for invertebrates, but not so good for bigger vertebrates, like your mega herbivores that you have here. So you'd select different genes from those. And developing the right metabarcoding primer for your target taxa. So here's just an example of an earlier development, so the BF2BR2 primer looking at aquatic invertebrates, and what they found was only 23% of DNA was captured in the arthropod family, but then you had 55% in the diatoms, and so the diatoms are taking up a larger amount of your sequencing space. So developing a new metabarcoding primer that captures more of an arthropod-specific region is really good. Another one is databases. So there's incomplete databases for a lot of taxa out there, like arthropods, but there are movements like the International Barcode of Life Bioscan that aim to generate barcode coverage for over 2 million species by 2026. And commonly there's misidentification uploaded with the barcodes, so that can also give you incorrect results with some of these online reference databases. So it is important, if you're doing long-term monitoring, to develop custom reference databases. So a bit of a plug about my PhD. I'm looking at how to use invertebrates as bioindicators for monitoring ecosystem restoration. So here I've got a graph of ecological condition on the y-axis and time on the x. And pre-mining, you can see the ecological condition is quite good. And when mining occurs, it plummets. And then over time, you see during the rehabilitation stage, the ecological condition getting better and better. And ideally, what you want is a similarity to the reference ecosystems surrounding the mine. So here's an example of the Jabiluka mine site in the Northern Territory. In 2006, the mine was still there, and then it underwent rehabilitation, and you can see in 2018 it's slowly coming back. But there tends to be a bit of a vegetation bias with mine site or ecosystem restoration monitoring, and there's often short timescales for project completion. So as you can see here, like it's going to take decades, but sometimes they're only given 5 years. So Ranger Uranium Mine is also in the Northern Territory, and it's a large uranium mine that closed in 2021 and is currently undergoing rehabilitation and is due to be rehabbed by 2026. So what they want to know is how they can use invertebrates for part of their closure criteria. So ants and termites have been identified as bioindicators for this mine site, and they want to look at species composition, richness and abundance, and they want to make sure that all of these taxa are similar or on a trajectory towards a reference ecosystem. So I work closely with the federal government, called the supervising scientist, that are funding my project to look at all of these genetic questions for monitoring invertebrates. So I'm going to skip through because I'm running out of time, but my project is looking at how to address a few of these issues that I highlighted earlier. So one of the studies I'm looking at is a foundational study assessing the use of eDNA in soil substrates to monitor ant assemblages. So I'll be looking at putting some ants on some soil and then collecting the soil and seeing how long the DNA can last in the environment over time. So you just want to see, is it going to last a day, 2 days, a week, a month? We're not sure yet. Another chapter of mine is developing an eDNA and metabarcoding protocol for termite baits in the Australian seasonal tropics. So termites are quite hard to sample, and one of the methods is to use a baiting system, which you may have heard on the Monday talks. But basically, the termites can come and eat the bait, and they can leave, and they leave a small section of the bait still intact sometimes. And for the ones that do, I want to measure what DNA is there, how long does it take to degrade, and can we pick up termite species composition from that bait. So in conclusion, genomics approaches are a powerful tool and increases accessibility for managers and academics to monitor invertebrates. There are widespread applications including rapid biodiversity surveys, land use impacts, and monitoring ecosystem restoration. I just want to give a shout out to my supervisors and advisors and at CDU and across the federal government supervising scientists, and also to some of the traditional owners that work on the field sites with me. So my field work is conducted in the Narberlake mine in West Arnhem Land, and I just want to thank the Binning people for welcoming me there, and a bit of my contact information. And if you have any questions, please feel free to come up to me. I'm a huge genetics nerd, so ask away. Be happy to talk. Thank you.
Okay, so it seems like Frode is not here. So if anyone has a question for Alison, maybe one question?
Sorry.
Thanks to Frode. Does anyone have a question?
Thanks, Alison. Since you went in front, I will ask my question. On the graph where you had, on the y-axis, I think you had ecosystem...
Ecological condition.
That's the word. What was used as a metric for ecosystem? Was it the vegetation?
Yeah, normally. So that paper did deal with the vegetation only. It was just more of a schematic showing, how it does degrade, like a theoretical, how mining impacts affect the environment or the ecological condition.
Okay, so it's just like if the vegetation recovers, they think everything's restored?
Yes.
So that was the metric. Okay,
yeah.
Cool, thanks.
We had one.
So I was just wondering if, you mentioned the costs are coming down. The question is it now cheaper to use these methods than to send a field biologist into the field to do a biodiversity survey?
Well, yes and no. So the cost is coming down, but the initial validation surveys to set up a long-term monitoring barcode library can be a bit expensive because you still need the specialist taxonomic expertise to say this is this species, etc., before you then can move into the DNA workflow. But if you take, say, 300 pitfall traps or 1,000 pitfall traps, it may take 6 months for someone to analyse. That's a long, labour is a high cost. Whereas if you take the genomics approach, say you still need to get out the taxa that you're looking for from the pitfall traps, it may take a month, but then you can get results in a few weeks. So it shortens the time scale, and once those initial validation surveys are done, then it will be cheaper. Yeah.
Okay, thank you, Edison, for the great talk. I just want to remind everyone that we have a poster for this session by Dan Parker, titled 3 Decades Later in the Northern Kruger National Park, Snapshot Surveys May Underestimate True Local Richness of Bats. So we can check it out later in the afternoon outside the foyer. Our next speaker is Nikisha Singh. She's the curator of the in-house biological evidence collection, and she's going to be telling us more about that.
Okay. Good morning, everyone. My name is Nikisha Singh. As Kensani introduced me, I currently manage the Skukuza Biological Reference Collection. Okay. So the reference collection serves as an invaluable and indispensable asset to the Kruger National Park, and I'm just going to give you a bit of history. The reference collection started in the 1950s when Mr. H.P. van der Schyff started the first formal plant collections in 1951. He collected approximately 4,000 specimens, which are still housed in the Skukuza Herbarium today. The faunal surveys took place from 1959 until 1970 by Dr. U. de V. PNR, and this saw the addition of the zoological specimens to the current collection. This included numerous taxa, such as amphibians, reptiles, birds, small mammals, and other invertebrates. Okay. So the current zoological collection consists of the following: approximately 10,000 insect specimens, 1,257 reptile specimens, 487 amphibian specimens, 832 bird specimens, 129 mammal specimens, and more than 950 fish specimens. All in all, we have more than 25,000 biological specimens within the collection. Okay, so the Skukuza Museum and Herbarium the facility materialized in 1959 when Dr. Rocco Nobel laid the first cornerstone of the building. Since then, the facility has served many important functions, one being an identification service. So whether it be researchers, interested persons, or colleagues, we do offer identification services. So if there's a plant, an animal, or a bug that you might be interested in and it occurs in the Kruger National Park, we can assist you with identifications and information relating to that. Okay. The Kruger National Park has a very extensive natural history. This is owing to the extensive biodiversity that occurs within the park, and this biodiversity needs to be documented in order to look at specific trends such as population or population increases, decreases, distributions, etc., because these eventually do filter into management decisions and so on. So we also keep an inventory of all the fauna and flora that occur within the park. Okay. Over the years, we've had numerous national and international researchers that frequent the museum and the herbarium, and even so after COVID, we have got an influx of researchers. They use the specimens for anatomical and taxonomical studies to answer varied research questions. And one of the most important components, in my opinion, is the facilitation of education. We have many school and university groups that visit the museum, so they're able to expand not only their practical skills, but their theory on biodiversity, natural history, biology, etc., and the importance of conserving and preserving this. Okay. Databases and data management. With so many specimens within the collection, we need to have in place a database in order to manage all of this information. Okay. So we currently have two main databases. Our zoological data is stored on Access databases, while the botanical data is stored on BRAM 7. BRAM 7 is just basically botanical records and herbarium management system. Okay. In 2022, we embarked on the Herbarium Data Project. This included the 16,000 botanical records which make up our herbarium database. many colleagues assisted with drawing up protocols and procedures that were followed in order to clean this database, update taxonomic information, and standardize the data. For example, a lot of the locality names within Kruger have changed, so this needed to be updated, GPS coordinates need to be corrected, and so forth. Various other sources were also used to update the taxonomy, such as the South African National Plant Checklist, Plants of Southern Africa, and various IUCN red data lists. Okay. The future of the reference collection. Okay. So the future is digitization, but what exactly is digitization in terms of collections? Simply put, digitization is putting something into a digital format and having that available on an online platform that can be accessible to anybody that needs that data. With regard to specimens, this would be taking images of a herbarium specimen or a zoological specimen and having that data also available on an online platform with its associated bioecological data. Okay. So why should collections be digitized? Well, not everyone can afford to come to the Kruger National Park and experience our lovely collections. So having this on a digital database would mean that anyone across the world would be able to access this data and perform more research. Okay. And what are the benefits? Besides accessibility, if there were a natural disaster like we've experienced recently with the flooding in Kruger, and if the collection had to deteriorate or be wiped out by a fire or a flood, we would still have the data available for research to go on. Okay? And having this data available means that the physical specimens are not handled as often, so the physical integrity of the actual specimens can remain viable for a longer period of time. Okay. This year, we will be embarking on the Skukuza Herbarium digitization project. So all 16,000 pressed plant specimens will be imaged and put onto a database. We expect that this will take the next 3 to 5 years to complete. Specific workflows and protocols have been drawn up so that each step can be followed accordingly to minimize any errors and ensure that the highest quality data and images will be available. The goal, again, is accessibility. Okay. Just a bit of what we will be doing. One of the main steps in the main workflow is the pre-digitization workflow. So if we were to work with a batch of specimens, the first step would be to go through each pressed plant specimen and check if there are any repairs that need to be done. For example, if a leaf or flower or even a label were to be loose, you would first fix that. And then once that is done, we would update the taxonomy. One can refer to our database that has been updated to now update a genus or a species or a variety, and then we'd print out a little label with the date, and that would be stuck onto the specimen just to show the date when this was updated. Lastly, we would then attach a unique identifier, and that would just make up the pre-digitization workflow. And then this is just the main workflow. After the pre-curation digitization, the next step would be imaging the specimens. Many herbaria and many museums use different methods to image specimens. It just depends on the time available, resources, and staff. One could use a high quality scanner if funds are available, or a simpler method would be to use a camera stand and a sort of stand that your images would be placed on, and we will be using that in conjunction with the light box to image our specimens. So once imaging is done, batch of images will be imported into Adobe Lightroom Classic, where the editing will be done according to specific standards, and this will be exported to a hard drive and cloud just for storage until it gets to the database. Okay, here are just a few pictures as I was speaking of different workflows. You can have scanners, a workflow, a workbench flow method, or just a simple stand in order to get your digitization done. Okay. Thank you for listening. Sorry. Just a quick note, if you would like to come and have a look at our collections, we will have a tour from 7 to 7:30 tomorrow, or if you would like to know more about the collections, you can have a chat with me anytime I'm available.
Okay. Oh, there's a question for you.
Thanks for the talk.
Welcome.
I just wanted to find out, since you already, a lot of researchers come and use these collections, and some of them have got, better sensing equipment, you can use some laser to actually get some three-dimensional aspects. Is there any, since now you're going to have a barcode, or DOI, is there a way to encourage or actually demand that people who've actually have collected data use the data from your reference collection, it links up, they have to link up some, have uploaded to some repository that can be cross-referenced to your actual thing, so that now it actually makes it better for someone else, so that they don't actually have to come in, they can just work on those cross-referenced images, because that's where most of the data will lie.
I think for future, as sort of a future plan to collate all of that data and possibly have it available, we could look into that. Yeah.
Hi, Nikisha. Where are you going to store these eventually? The digitalized?
So the herbarium specimens and the data will eventually be available on a database, but that's still in progress.
Okay, you haven't chosen a database.
So we're in collaboration with SANBI, but I'm not sure when that database will actually be available to everyone, but that will be the end goal.
Okay, the actual images, SANBI uses JSTOR, and various other herbariums, South Africa, have put all of these into JSTOR plants, which is obviously internationally freely accessible to anybody. There's incredible stuff there.
I've seen it.
I don't know, have you? Yeah.
Yes, I've seen it. It's great, actually.
I was just wondering if that's your end goal.
That will be the eventual end goal. It will just take some time to get there because, getting everything imported to the databases and clearing, whatever needs to be
and their
done.
standards.
Yeah.
We're still good on time. If there's one more question. Okay.
Thank you. Is it possible to upload your own collection? We're part of the Greater Kruger, so not the Kruger itself, but we've got an insect collection.
Of the Greater Kruger. I mean, we could chat more after this and then look into that.
Thank you, Nikisha. So with that, we're going to transition into Yose's solo session. His first talk is titled Summarizing Cold EMS Results on Land Surface Dynamics in Pristine Environments. Thank you, Yose.
Yes, ladies and gentlemen, good morning still. Thank you to you that you're here. I actually thought that this would be a talk which is at the front of other contributions which you listened to in this session already today. You as well learned from Chris earlier on what Cold EMS stands for. It's the contribution of these BMBF Spaces projects, SALDI and EMS Africa, two projects which are ending this or the next month, unfortunately. The aim of Cold EMS was to monitor and model land surface dynamics in areas with currently low human impact across a climate gradient in South Africa, and Chris has shown some animations about it. Chris mentioned as well our 6 study sites, of whom 4 study sites incorporate sandparks areas. That's in the southwest, the Agulas National Park in our Overberg region, then Ogravis Falls, Mokala National Park, although there we actually changed to Benfontein Nature Reserve, and the final thing is Kruger National Park, where we have been working already for quite a long time. I mentioned that there were several contributions of Cold AMS to this Savanna Science Network meeting. Parallel to my talk, there is a poster on gully erosion in Mokala National Park, and we have two other posters, one on the Zaldi data cube, which Chris mentioned, and then Marco's work on woody cover assessment in KNP. And that probably shows that there's a lot of potential for discussion, as we saw this other woody cover assessment presentation in this session here. Yeah, this is just a teaser to come to my poster, UAV-based survey of gully restoration in the Mokala National Park, which is a rather young national park building up on other land use types before being a protected area. According to the very short time I have, I would like to show you here one of our results, because what we did was we revised the RUSLE soil loss assessments, which have been done by Leroux et al. 2008, now 15 years later, with different input data. And if we look at the Kruger Park, which you can identify here in the upper northeastern corner of our Echlanseni site, then you can see the color coding is the same, that there is a big difference between the assessments. J. Leroux and others assessed something like 8 tons per hectare and year of soil loss, and we come up with 0.3 tons per hectare and year, which is definitely below what many people call tolerable soil loss. So obviously, soil loss in the Kruger Park area, as well as in the whole of Echlanseni, is not as bad as it was thought to be. And if we compare our assessment with values which I derived 2018 from reservoir siltation studies in Kruger Park, then we can see that we are more closer according to the order of magnitude than the previous assessment of soil erosion for this area. And we've observed that in any of our other 5 study areas as well, that there is a considerable difference between these two assessments. And that is actually what I wanted to tell you here shortly. Thank you for your attention. We have to thank for the Federal Ministry of Education and Research for funding our projects over the past, initially 3 years, then it became 5 years because of COVID, and certainly for the support of Sandpark Scientific Services, section rangers, game guards, and other helpers. Thank you so much for your attention. Thanks.
Okay, thank you. We can take one question for Yosei. Okay, thank you.
We can talk outside when we have wine and cheese later on.
So we're going to move along to our next session, the response to rainfall gradients and drought. And our next speaker is Sally with her talk titled Tree and Grass Drought Responses in Seasonal Arid Ecosystem. Thank you.
Hi everyone, it's great to be here, and my apologies. I've changed the title somewhat. Is it coming up? I've changed the title somewhat, but I'm going to be talking about the same things. Okay, so I'm actually going to be focusing a little bit more on grasses. We're going to chat about trees over tea, and we're going to talk about mid-season droughts as drivers of grassland functional types. So just to sort of set the scene, we know that the best really prediction of where you're going to find savannas globally is the fact that savannas tend to have this very, very predictable 3 to 9 month drought every year. And we know that it drives the ecology of the system. We bring it into our thinking and our science. We also know that many savannas are exposed to occasional multi-annual droughts. There's less work on this, but we also recognize that it's a very important driver of the system. However, many of these savanna ecosystems are frequently exposed to 2 to 3 weeks of no rain in the middle of the growing season. And the importance of these mid-growing season droughts has not really been recognized. So I'm going to be sort of raising some questions around how mid-season droughts have shaped and constrained adaptations to seasonal and multi-annual droughts. Because anyone who delves into the drought literature, one of the hardest things dealing with it is people say this is a drought adaptation, but the types of adaptations you'd need to cope with a 3-week drought is very different from what you'd need to cope with a 3-year drought. So how do these different evolutionary drivers interact? So Ben has alluded to this already, that Kruger's a wonderful example of places where you can get 6 weeks of no rain in the middle of the growing season when the plants are all hyped and ready to be active. So let's kind of challenge our conceptual theoretical model about how seasonally arid systems work. In theory, you have alternating growing and dormant seasons. Anyone who's looked at a soil moisture data set knows that in reality you have these fluctuating soil moisture values with various periodicities. There's definitely a seasonal cycle, but there's also sort of finer scale variation. But then from a plant's perspective, there's either moisture in the soil to grow or there isn't, and that is controlled by the soil texture. But if you kind of work out what is the soil moisture level for the soil texture, where plants wilt, the wilting point of the soil, which is like minus 1.6 megapascals, people think, then you can like draw the solid line through that time series, and you end up with a data set which is really just a frequency distribution of periods of plant growth and plant dormancy. And in this data set, which is from the Flux Tower in Skukuza, we can see that sometimes those periods are several months and sometimes just several weeks. So with my PhD student, Mthokozisi Moyo, he's produced a data set of plant growth days. So he's taken a soil moisture data set, overlaid it with a soil texture data set, worked out where the wilting point is, and classified whether there's soil available for growth, there's water available for growth on this day in this part of Africa. And you can see that some savannas behave very predictably. So that's a time series from Nyasa National Park where you have seasonal rainfall, but other savannas like Kruger have very different sort of growth day occurrences through the years. So we've taken a couple of sort of classic parts of Africa, basically places I've been to and understand, and I've drawn the frequency distribution of growth of dry spells. So basically I've just quantified the number of days where plants can't grow, and I've created a frequency distribution of that. So going from lowest rainfall environments like the Northern Cape, Tswalu Nature Reserve, all the way up to Lope in Gabon with 1,600 millimeters of rainfall, you can see that while there are some savannas that behave as you would expect, so in Zambia and Mozambique, you basically have a lot of clusters around that 200, so that's like a dry spell of 200 days, so a dry season. And then occasionally you have some dry spells during the growth season of a few days, like 5 to 10 days. So that's the signal you'd expect for a seasonally arid system. Zambia and Mozambique and Madagascar fit what our concept of how savannas work. But many other savannas, especially the arid savannas, you end up with, can I point? No, I can't point. So you end up with like very high frequencies of dry spells in the sort of 10 to 30 day time period. So plants during the growing season are often exposed to 10 to 30 years of dry spells. And you'll see that although, savannas in the wet savannas seem to behave more like how we think seasonal systems should behave, you can get really wet places like the Drakensberg that still have frequent dry spells. And if this way isn't a nice way to think about it, you can sort of quantify it as the probability of having a dry spell of different periods. So a value of 1 means every year you have a dry spell of that period. So a dry spell of 60 days occurs in Madagascar every year, predictably. It basically has a seasonal drought. But like the Serengeti, which is a bimodal rainfall system, hardly ever has more than 60 days of dry concurrently. And places like Impala, Kenya, often have like at least 2 periods a year of more than 60 days of dry. So it's like a way to sort of characterize how long the plants are exposed to drought. So as I've said, some of the savannas that we think about they behave how we expect them to. Others actually have quite different kind of periodicities of dry spells. So under that condition, now that we know not all savannas are the same, well, how should a clever plant grow? Norman Owen Smith wrote a paper several decades ago asking what clever ungulates should do. But perhaps a more appropriate way to ask the question is what water use strategy should be selected for in systems with these different types of droughts. So to address that, I'm just going to sort of make sure we're all on the same page in terms of 2 important concepts. The first one is the safety efficiency trade-off. I put that figure in for the physiologists who know about it, but it's kind of, for me, the ecologist, it's just a simple question of when exposed to drought stress, an organism, a plant, can close its stomates to conserve water, but then they're sacrificing carbon gain. Alternatively, they can just keep pumping away and photosynthesize, but then they run the risk of cavitation, hydraulic failure, and loss of above-ground biomass. So that's a classic trade-off in the drought literature. The second concept I want to talk about is something that the trait people, Salguero Gomez, calls shrinkage. And this is a fabulous paper from PNAS where he shows that shrinkage is actually a really important sort of major axis of variation that describes plant life history strategies globally. For a person who thinks about grass and a grassland ecologist, shrinkage is a very, we think about curing all the time. So grasses, when they cure, they're basically just shrinking. So for people in our ecosystems, shrinkage is something we think about and work with all the time. They just use this term. Many trees also, so top kill is excessive shrinkage. You could think of it that way. But for a grass, which is quite a simple organism, which has no solid above ground biomass, shrinkage is the same as top kill, is the same as curing, is really the same as deciduousness. So it's like how much above ground leaf area gets lost. And it's, as Salguero Gomez shows, it's often really adaptive to be able to shrink. So what would the expected strategy for a grass be in a classically seasonal environment? Well, this is how modelers tend to model grasses, and I know I've published a model where I basically modeled grasses growing like this. The rain starts, you put on your leaves as quickly as you can, you keep your leaves, the rain stops, you carry on photosynthesizing until you run out of water, and then you cure. So you basically cure, your leaf area just responds facultatively to rainfall. However, if you have that strategy and you're exposed to a seasonal drought, you put on your leaves, you're growing like crazy, the drought comes, you keep transpiring, you start to shrink. By the time the rain comes again, you've lost all your above ground leaf area, you have to put it back on again. So it ends up not really being particularly adaptive strategy if you're exposed to long seasonal droughts, because you're losing carbon by having to take time to regrow all that leaf area. So theoretically, in a system exposed to mid-season droughts, the alternative strategy, the safety strategy, might actually make more sense. You put on your leaf area at the beginning of the growing season, and then when you get exposed to drought stress, close your stomates, reduce water loss, reduce carbon gain, but maintain that green leafy canopy. So when the rain comes again, you just carry on going. You're sort of sailing through that mid-season drought. So theoretically, that should be a better strategy in places that have mid-season droughts. Okay, so what have we discussed so far? I've shown you that mid-season droughts, up to 4 to 6 weeks, as Ben showed, are common in African savannas. And I've sort of suggested that predictable growing seasons should select for efficiency and rapid curing in response to drought, whereas mid-season droughts should select for safety and maintaining a functional green canopy for as long as possible. So I suppose the next question, for however much time I have, is to check, do we see any evidence for these strategies in our systems? The modelers tend to think of grass, we tend to just model grass as one thing, and the C4 grass follows that efficiency strategy. Like, is there any evidence that we're kind of missing some important variation in our ecosystems? And then if so, what are the ecosystem implications of that? So in order to look for evidence of these strategies, we set up a wonderfully huge greenhouse experiment with my student Londiwe Mokoena, who's there in the corner. We had 15 different grass species that we chose because we thought they might have different drought strategies. We imposed a 60-day drought. We measured stomatal conductance, soil and plant water potential, osmotic adjustment, and curing, or shrinkage. And then we also looked at recovery after drought. Did they rehydrate? Were their canopies still active? Or did they have to resprout from the base? Did they have to regrow a leaf area? So what did we find? We certainly found a range of curing rates. So on the x-axis, you have soil moisture. So as the soil moisture decreases, on the y-axis, you can't see it. That axis is the fraction of green leaves. So how much of the canopy is green? And of course, as the soil moisture declines, plants cure, the fraction of green leaves decreases. But some plants, especially Panicum maximum, as you could see, had massively higher rates of curing for the same water stress. So we do see a range of curing rates under the same drought. What about the drought response strategy? So here on the x-axis, I have plant water potential, which shows like how drought stressed is the plant. And on the y-axis, we have stomatal conductance. So again, we find that across those 15 species, for the same water stress, some plants will have totally closed their stomates, conserving water, and other plants will keep their stomates open, keep transpiring, basically expose themselves to cavitation following the efficiency strategy. So we do find variation in grasses. It's not appropriate to think of them as just one functional group. But the real question is the drought response causing the curing rates? I set it up as implying that because the plants are closing their stomates, they keep their canopies green. Is there any evidence that the stomatal control drives the amount of leaf area you can support? So we have some limited evidence for that. It's not a particularly strong relationship. It is significant. So on the x-axis, we have the water potential when they close their stomates. So how stressed were they when they decided, no, I'm not going to keep photosynthesizing? And on the y-axis, we have leaf moisture content, so how green was the canopy? So there's some limited evidence that this trade-off occurs, which is exciting, but my master's student will be going into that more in her paper. I just wanted to spend the last little while talking about the bigger implications of this, the ecosystem implications and the biogeographic implications. So to look at that, we set up this lovely big experiment with my colleague Michelle Griewe at the Pretoria Experimental Farm, where we put these different grasses with different drought strategies in like a community ecology experiment, so sometimes different mixtures of species. And we grew them, they were all very happy, and then we exposed them to a drought. So you can see that at the end of a drought, we have some communities that are still very green and some that are pretty much cured, and then we set fire to that. So next year, I'm going to come and tell you whether the drought response strategy of the grasses drives the fire regimes. That's our hypothesis, that cliffhangers, it's the way to go. And I'd really love to hear some comments about this.
How are we on time? We can take one question for Sally. Okay.
I tried to politely hesitate to see if anybody else had a question, but I'm dying to ask. How much do you think it matters? I'm not quite sure how to phrase this, but I could imagine one strategy would be beneficial if you knew that the dry spell was going to be a short dry spell, but that another strategy might be beneficial if there was the possibility that the short dry spell would turn into a protracted drought. And I would imagine that the optimal strategy might differ in those two scenarios, but that a grass may not know which one of those things is going to happen. And how do you balance that sort of basically a lack of information problem? How do you think about that?
Exactly. So these really kludgy figures that I put here were the beginning. I do actually have a model on this that isn't perfect yet, so I can't present it, because we'd have to do a little sensitivity analysis with different dry season lengths. But it would be fun to chat to people about what they think about which of these dry season lengths are the one, where's the threshold where the strategy should switch? I think that's your question. And I don't know, but we could model it. Shall we model it together? I need some input on the model.
Thank you, Sally. Oh, there's one question there.
Yeah, it's the problem with this trait work is these plants are evolving under lots of stresses. The way we've thought about it the most is, potentially the plants become less flammable when they follow the safety strategy, but they might also become more palatable because one way to maintain a green leaf canopy is to osmotically adjust, which means you put salts into your leaves. I was chatting about this with a chemical ecologist last week. So that's an idea that my colleague gave me. So we've thought more around. We haven't really thought about how resistance against herbivory affects this, but we have thought around whether drought strategy interacts with palatability.
So have you thought a bit about temperature? So if you're going to have these extended droughts, what is the effect of different daytime temperatures? And surely that's also going to affect what the grass is going to be doing.
Yeah, thank you for asking that question. If I'd had more time, I would have tried to define the growing season using temperature and then looked at the probability of droughts during the growing season. Because some of these short droughts actually happen in the dry season, in which case, and the cold season, in which case the plants won't be growing. So it should be possible to address that. I just haven't done it for this talk, but thanks for bringing it up. Yeah.
Okay, moving along. Our next speaker is Rob, and his talk is titled Precipitation Gradients Drive High Turnover in the World's Largest Savanna. Thank you, Rob.
Hi everyone, I'm here to talk about part of my PhD research, which is looking at how biodiversity of savannas is structured, particularly in the southern African woodlands. Before I start, I'd just like to draw attention to this long author list I have, which is broadly as a result of my part in the CSR network, and I know a lot of these people are here at the moment, and I'd just like to thank them for their contribution to this network of data. So when I think about the biodiversity of these ecosystems, I'm going to deal with three different facets of that biodiversity. Firstly, species richness, secondly, beta diversity, and finally, phylogenetic alpha and beta diversity. So before I get into that, I'm just going to give you a bit of a background on biogeography. So we know there's this large variation in the distribution of global biodiversity. And we see these hotspots of biodiversity, particularly around the tropics. This is a graph of a map of vascular plant richness. And we get these hotspots of biodiversity in the tropics, and as we move polewards, we kind of move down this gradient of richness. And whilst global biodiversity isn't distributed equally, it is distributed in seemingly predictable ways. And one of the strongest patterns that we see is this covariation and strong relationship between climate and taxonomic diversity. So this is a different map of global annual precipitation, and we see a largely similar pattern here whereby the tropics are very wet and we get this drying as we move polewards. And when we combine this rainfall data with temperature data, we actually find really high covariance, and when in combination, they determine up to 70 to 90% of the variation in diversity. And these positive relationships of richness and climate are observed across taxon regions. So this first graph on the left-hand side is from the northwestern South America, and we see an increase in the tree diversity as we get declining water deficit. And the same pattern is seen for birds globally. We also see this clear relationship between climate and phylogenetic diversity. So phylogenetic diversity is a quantification of the total evolutionary history of a community. It's quite often given in this PD metric, which is a sum of the branch lengths of a phylogenetic dendrogram. But we do see slightly different patterns when we consider phylogenetic diversity to when we look at alpha diversity. So this study here from lowland South America shows a peak in phylogenetic diversity at intermediate rainfall of about 1,500 millimeters. And what this is caused by communities at intermediate rainfalls containing lineages from both species found in the low rainfall areas and those in the high rainfall areas. And understanding how these patterns play out within other ecosystems is important, given the role of phylogenetic diversity in maintaining ecosystem services and functioning. Also, so we know these communities are highly structured over these patterns, but why do these patterns exist? And one of the reasons for this could be that species have a differential ability to deal with these climate stresses that we see. Again, this is a study from Panama, and it shows that those trees that we find that are really drought sensitive are found in much higher densities in wetter forests. We also know that climatic extremes of temperature, drought, frost, they place limits on the ranges where we expect to find these species, and that species that are more resilient to these stresses are likely to be more widely distributed, while those with narrow tolerances are less likely to be as widely distributed. And this can play out in two different ways when we consider beta diversity. So we could see a pattern of nestedness, which, if you look at panel A on the figure, shows that in these less diverse, drier areas, we would find a subset of the species that we find in the most diverse, wetter areas. Alternatively, we could see a pattern of turnover, which, if you look at panel B, this is where species that are replaced by wet or dry adapted species that are not present in the areas with more or less rainfall. And we have different patterns of this in different environments. So in Panama, we see large turnover across rainfall gradients, whereas in South America, there's clear nestedness. But the bulk of research on these patterns has been focused on the moist tropics, and we don't know whether these same patterns are seen in savannas. We know savannas are really different to moist forests. They experience high levels of disturbance from megaherbivores and fire. And while we know that precipitation and these disturbance drivers are important determinants of the structure and distribution of savannas, how they play out in broad compositional patterns of savannas is less well known. Some previous studies that were foundational to energy theory, energy richness theory, did look at this in South Africa, but these were based on herbarium data and quite a coarse scale. So these blocks are placed over South Africa, so they cover quite a big distance. They did find a positive relationship though, but we really lack the plot level data currently to assess these relationships. So as part of my PhD, I aim to resolve this by looking at how the savanna tree communities of the southern African woodlands are structured over these broad environmental and disturbance gradients. As I alluded to before, I'll do this using three separate facets of biodiversity. Firstly, tree species richness, beta diversity, and finally, phylogenetic alpha and beta diversity. And this is all largely thanks to the CSL network, which is, there's a poster on that outside in the foyer, actually, presented by Lorena, if anyone's interested to learn more. It's a network of researchers from the region and brings together lots of data to help understand how these woodlands are changing. And it spans 9 countries across the region, and because of this I benefit from a really large, really wonderful data set of over 150,000 stems and over 6,500 plots. A lot of these plots are different sizes, so I constrain my analysis to just focus on those that are between 0.1 and a hectare, and also filtered them down to just be the ones within White's classification of woodland, as I'm focusing on tree species and those with over 10 centimetre dBH. The range of the data where the plots are gives me this really nice gradient of temperature that spans almost 10 degrees Celsius change, and also a really wide rainfall gradient from about 350 millimetres up to almost 2,000 millimetres of rainfall. So to assess how the biodiversity changes, I used a negative binomial model because we had some over-dispersed count data, and I inputted all these predictors that were from a range of sources, so the climate data from WorldClim, soil grids for some of the soil data, and others. I also looked at beta diversity patterns using Sorensen dissimilarity. So this is comprised of 2 separate components, and you can tease these apart. The first of which is Simpson's beta diversity, which gives a measure of turnover, and what's left over is nestedness. I then used generalized dissimilarity modeling, and this allows us to identify which of the predictors that we place in this model are driving either the turnover or nestedness that we see playing out in the beta diversity patterns. So on to some results now. This first, considering species richness. So this is a coefficient plot from the model I ran. On the bottom is the effect size. Up the y-axis is the predictors within the model. Points to the right of the dotted line, a positive effect. Points to the left are a negative effect on species richness. And what we really find is that the wetter savannas contain more tree species, and that rainfall is the largest driver of changes in species richness. Rather surprisingly, I guess, for savannas, there's very little, or there seemed to be very little effect in the data I looked at of soil, herbivory, and fire on the species richness of these savannas. These are then conditional effect plots from the same model, and what these do is they hold all the other model predictors at their mean value and then plot just the effect of the chosen predictor. This is just to show a difference in the amplitude of the effect. So precipitation is panel A, and that shows a strong increase as we move up the rainfall gradient. A mean annual temperature, which was our third largest effect size, has this almost flat trajectory over its entire range within our data set. So secondly, I looked at beta diversity and how that's structured across these gradients. And what we found was this overwhelming turnover in species as we moved across our gradients. And that's characterized by this beta Simpson's value of almost 1. And that, if you remember back to the original slide, the first slide on beta diversity, this turnover in species. So at each plot, we're finding almost a unique composition compared to the plot before it. And then our generalized dissimilarity model allows us to tease apart the predictors that drive this turnover that we see. So these are I-spline plots from that model. They're a bit confusing, but the take-home message is that the higher the bar, the higher the line, the more important that predictor was in driving the beta diversity pattern that we observed. So at first we have seasonality, mean annual precipitation, the distance between plots, and then finally the temperature, the mean annual temperature. And what we found is that these beta diversity patterns are largely driven by the precipitation regimes that these plots undergo. First, primarily seasonality, followed by volume. And the effect of these were particularly pronounced in between the low rainfall sites and up to those with intermediate rainfall. And finally, considering phylogenetic diversity, this first one, we largely found the same results that we saw for species richness analyses, and that's not that surprising given that the R squared of PD and species richness is sometimes up to 0.99, so we expect to see these same patterns. But when we considered phylogenetic beta diversity, what we see is a slightly lower turnover than what we found with taxonomic diversity, and then a greater phylogenetic nestedness, which is more like panel C that you see at the bottom of this figure here. And again, these are just a couple more ice-blind plots from the phylogenetic GDM that we ran. And again, precipitation seasonality was the most important predictor of phylogenetic beta diversity. But interestingly, fire count at these plots started to have a greater effect on the phylogenetic beta diversity patterns between these communities, especially where we saw this high fire, highly flammable plots. And that could indicate that fire is having a filtering effect on the phylogenetic diversity that we see at these plots. So just to summarize, in these wetter savanna woodlands, we see much more tree species as we move into the wetter areas, and that is also seen with greater evolutionary diversity. And we see this really large turnover across this big region, and this turnover was largely driven by the precipitation regimes. And as I move forward, I'd like to consider how this plays out in functional traits. So we see that precipitation is driving these biodiversity patterns. And is this because of the differential ability of species to survive these precipitation gradients? And also, can we then use these traits to understand the mechanisms that drive these patterns that we see? So is it a result of there just being more trees where we see more rainfall and therefore you get more species? Or is it that more species have the ability to survive in these wetter environments given that they evolved in wet environments, and that fewer species actually have the ability to move down the rainfall gradient into these drier savannas? So with that, I'd like to thank you all for listening, and again thank the CSL network and others for their help in doing this research. Thank you.
Thank you, Rob. Any questions for Rob? Okay, thank you. So we're going to move along to the water, carbon, and energy flux session. Just waiting for people to move out of the room. So we're going to start with a 5-minute talk by Oksana, and it's titled Biophysical Control of Evapotranspiration in Semi-Arid Karoo and Savanna Ecosystems.
Yes. Hi everybody, my name is Oksana and I totally love microphones. So today I'm going to present you my research on biophysical controls on evapotranspiration in South African, Karoo and Savanna ecosystems. And first I'm going to present my study sites. So we have currently working with 4 study sites, which has paired approach of eddy-covariance measurements. So the first 2 sites are located 1.5 kilometers far from each other in the Middelburg, and it represents 2 ecosystems, one with a linear grazing, another with experienced grazing. And by that I mean heavy grazing following the South African standards. Another 2 sites, it's Benford sites, which represent different biomes, Namakro and Savanna biomes, located in the nature reserve. Here you can see also pictures of all our measurement towers. First of all, I'm going to start with temporal dynamics of evapotranspiration, and what we can see at this figure, it's evapotranspiration, daily evapotranspiration with green and blue points for the Middleburg sites and black and yellow for the Benfontein sites. First, we can see it's clear pattern of the evapotranspiration seasonality, which clearly depends on precipitation and its distribution throughout the year. But also we have here mean evapotranspiration like for monthly, so we can compare different sites, Benfontein and Middleburg. And as for Middleburg sites, November, December months, we have only transition months, not yet wet season. While for the Benfontein sites, we already have growing season, start of the growing season, and September and October represents transition phase. Water use efficiency also quite different between the sites, with a light difference between the Middleburg sites and slightly higher difference between the Benfontein sites, which clearly shows that we have slightly different climatic conditions at the Middleburg and Benfontein sites, as well as different vegetation types and different root system, which raises the difference between the sites. So here we can see temporal dynamics of evapotranspiration, but monthly cumulative and association with precipitation at the sites, which you can see in the gray pattern. As we can see that difference between the sites, between the Middleburg sites, for example, under the under panel, is minimal and mostly driven with slightly higher precipitation at the sites. Why we can see a slightly higher difference between the Benfontein sites, which mostly driven by different vegetation types as well as slightly higher precipitation. The difference between Middleburg and Benfontein sites are much higher, but it's explainable considering the difference in precipitation. Here we can see mean diurnal evapotranspiration versus from the left and net radiation and vapor pressure deficits from the right. What we can notice at first that we have clear differentiation between dry season, transition, and wet season. Although all four sites working quite similarly to each other during the dry season, and we see only tiny difference. Why difference rise much higher during the transition and wet seasons? I don't have much time, so I just will go further. So here we can see evapotranspiration versus precipitation, and all the sites have shown strong correlation, although it has different slopes, which basically show us a difference in water availability as well as response of the vegetation towards it. And at last I want to show you evapotranspiration versus net radiation and vapor pressure deficits. And this is a point where we will go further investigating details, because to find out the difference at what point the sites exhibit difference in response towards evapotranspiration fluxes. And at first it looks like they are not quite similar, those patterns for Middleburg and for Benfontein data. But this is only because it's different seasonality. So here it starts connections from the November months, while at the Benfontein sites we have start of the transition months at September. So if we just shifted 2 months earlier, it will look alike, except with higher evapotranspiration at the Benfontein sites, which basically due to the precipitation. And we notice like strong correlation with net radiation, while slightly weaker correlation with vapor pressure deficits. And basically that's it. Thank you for your attention.
Thank you, Oksana. Unfortunately, you're out of time. Our last speaker for this breakaway session is Amgelani, and he's going to be talking about an approach to compare carbon dioxide and water fluxes between two vegetation types.
Good afternoon, everyone. My name is Amkela Maluleke, and I'm presenting today on a chapter of my PhD, which is looking at a per-tower approach to compare CO2 and H2O fluxes between two vegetation types representing Savanna and Namakaru biomes. So firstly, a major focus of earth ecosystems and science has been understanding the behavior and vulnerability and resilience of carbon sinks and their responses to anthropogenic forcing such as climate change, urban expansion, and agriculture. With about 30% of anthropogenic carbon emissions taken up by terrestrial ecosystems, it becomes then important to understand how land use practices and management can kind of impact on our climate change mitigation objectives, especially as we're placing our ecosystems as critical in achieving net zero emissions. So our semi-arid ecosystems are showing some greening trends and driving the global interannual variability in global carbon cycle. So it's then important to quantify these possibilities on how then can our ecosystems help us achieve these net zero emission status. And this is not as easy because our ecosystems are driven by the limitation of water, where the role of water is quite pronounced in these ecosystems. So the trade-offs between water and carbon will remain a conundrum in how we then get to understand the behavior of these ecosystems, especially as they are quite diverse. And my research question is, we have, as Oksana just showed us, the study sites in the middle of South Africa in Benfontein. So we're presented with almost identical climatic conditions, and then the question is then how do the seasonal and interannual patterns of CO2 and H2O fluxes differ between these two vegetation types. The savanna is the tall tower there, which has a mixture of grasses and trees dominated by Schmidtia parphorides and Stepacrostis uniplimis with several tree species, namely Areolobas, Totuolis, Tessia lancia, and Senegala mellifera, while the Namakaru patch is dominated by Pensia globosa. So, firstly, just presenting on the fluxes between the two. So we're looking at cumulative carbon fluxes between the two sites, where the data set has been split into two periods. And for the first period, we had about 524 millimeters of rainfall compared to 405 during the second period. And as we have a look, here is the first period at the Savanna site, which saw an uptake of about 100 grams of CO2 per meter square, compared to the drier period, which this was almost half of the uptake in the first period. And during the first period at the Namakaru site, we saw about 46 grams compared to just 11 grams during the second period. And there was a fire that came through, which we kind of estimate around 932 grams of CO2, of carbon being lost from the fire in late September 2021. And this is compared against a mean uptake of about 78 grams of uptake per year at the Savanna site. And looking at the controls of water, we kind of see that under different soil moisture classes, the Savanna is much more productive during the intermediate period, so that's between 5 and 10% of soil moisture. And the Namakaru biome is much more productive when soil moisture is more than 10%. And then becomes a bit neutral during these soil moisture classes between 5 and 10%. So, and once then that tips below 5%, it really becomes a slight source of CO2, while the Savanna site kind of still maintains a relatively small sink status of CO2. And looking at the patterns between CO2 or carbon and evapotranspiration. So here I'm just looking at the water use efficiency for periods where the incoming solar radiation is above 200 watts per meter squared. And we are seeing these linear patterns between GPP and ET. But for higher GPP rates along similar ET rates, we see that the Savanna is actually having much more uptake, which then is indicated also in the water use efficiency between the two sites, which is much higher at the Savanna site compared to the Namakaru site. This is for overall, over the 2-year period of assessment, where we see that the Namakaru is actually 13% lower than the Savanna site in terms of the water use efficiencies. As I said, these are different values to what Oksana has just showed us, because she's looking at the whole period, including the nighttime data, and I'm just looking at periods where soil moisture is incoming radiation is above 200 watts per meter squared. And yeah, just in closing, Savanna site, much more productive, and quite a threefold difference between the two sites, which are under almost identical climatic conditions, and more carbon is sequestered during intermediate soil conditions at the Savanna site compared to the Namakaru, which in both systems, you see that this gradually decreases as soil moisture decreases, when the Namakaru, interestingly, becomes a slight source under 5% soil moisture conditions, with Savanna having high water use efficiency compared to the Namakaru, but all throughout this measurement period, both sides were net sinks during the monitoring period. And yeah, that's it. Thank you.
Okay, thank you, Amu. That's it. Thank you for the wonderful talk. You're out of time. So just a reminder again, we have 2 posters from this breakaway session, one by UC and another one by Dan, so please go check them out. And again, there's a planned visit to the Skukuza Herbarium planned for tomorrow morning, so you're encouraged to show up for that as well. Thank you, everyone. Enjoy your lunch.